Preface of the Flea. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. The Flea by Harold Russell. Preface. The aim of this book is to give in plain language some account of a small but noteworthy group of insects. I have avoided, whenever I could, using the technical terms of zoology. To avoid doing so entirely is impossible in a book which describes insects in some detail. No technical term has, I hope, been used without an explanation. Over 30 years have elapsed since Taschenberg's German book, Die Floh, appeared. Our knowledge has made enormous strides since then. More species of flea are now known from the British Islands alone than were ever then known from the whole world. So far as I am aware, no book devoted to what is known about fleas has ever been published in English. The statements about these insects in the general textbooks of etymology are frequently antiquated and inaccurate, but there is a fairly extensive literature on the Xiphornaptera, scattered through scientific periodicals mostly in English, German, Italian, Dutch, and Russian. I have given some references in the bibliography. The naturalists now living who have devoted any time to the special study of fleas may almost be counted on one's fingers. In England there are Mr. Charles Rothschild and Dr. Jordan; in the Shetland Islands, the Rev. James Watterson; in Germany, Taschenberg of Hall and Dampf of Königsberg; in Russia, Wagner of Kiev; in Holland, Udamans of Arnhem; in Italy, Tiraboschi of Rome; in the United States, Carl Baker, and a few others. I have not mentioned medical men who have investigated fleas in connection with plague. There are small collections of fleas in the Natural History Museums at South Kensington, London, Paris, Berlin, Königsberg, Vienna, Budapest, St. Petersburg, and Washington. Of private collections, Mr. Charles Rothschild at Tring is by far the best in the world, and contains something like 100,000 specimens, and is most admirably kept. I must express profound and sincere gratitude to Mr. Rothschild for having helped me in numberless ways and advised me in many difficulties. It is well known that the mere mention of fleas is not only considered a subject for merriment, but in some people produces, by subjective suggestion, violent irritation of the skin. The scientific study of fleas has, however, received a great impetus since it has been ascertained that they are the active agents in spreading plague. Rat fleas are of various kinds, and not all fleas will bite man. A knowledge of the different species has suddenly become useful. The humble but ridiculous systematist, with his glass tubes of alcohol for collecting fleas, his microscopic distinctions, and Latin nomenclature, has become a benefactor of humanity. Some people may seem to be practically immune to the bites of fleas, but even to such persons their visits are unwelcome. A famous Frenchwoman once declared, "Quittez-moi s'il n'est pas à la muscure est la promenade." H.R. London, September 1913. End of preface. Read by Elijah Fisher.
Chapter 1 of The Flea. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. The Flea by Harold Russell. Introductory. Fleas form a group of insects that have, until recently, been little studied by zoologists. We call them insects because they are jointed animals, or arthropods, with 3 pairs of legs in the adult condition. The reader will best understand the position which fleas occupy in the general classification of animals by remembering that the arthropods, or jointed animals, are one of a dozen subkingdoms, or phyla, to which the various members of the great animal kingdom have been assigned. There is good ground for believing that all the animals included in each phylum trace their ancestry back to a common primitive form which lived in more or less remote ages. Besides insects, the arthropods or jointed animals include crustaceans such as crabs, lobsters, shrimps, woodlice, water fleas, and barnacles, myriapods such as centipedes and millipedes, and arachnids such as spiders, scorpions, mites, and ticks. To all these varied forms of animal life, fleas and other insects are therefore more or less nearly related. The animals belonging to this large and important collection, which compose the arthropod phylum, have certain common characteristic features. We find a body made up of a series of more or less completely similar segments, placed one behind the other. In this, they resemble certain worms, which are far less highly organized. The body is elongated, symmetrical on either side, and the mouth and anus are at opposite ends. There is, however, an important advance on the segmented worms. Each typical segment carries a pair of appendages which are very different from the foot stumps that are found on certain worms. These appendages of arthropods are divisible into distinct limb segments, separated from one another by movable joints and acted upon by special muscles. The common ancestor of all the various arthropods which are found living on the earth today was probably composed of a series of segments, each very similar to the last, and each bearing a pair of very similar appendages. In the course of ages, these appendages have been astoundingly modified in form and in function. So it happens that we find in the arthropods of the present day pairs of antennae, of mandibles and other mouth parts, of pincers, of legs, of swimming feet, and of tail pieces, which on close examination can all be traced back to a common structure. The body segments also have been strangely fused together and modified. All that has been so far said applies equally to fleas and to other insects. It is of great interest, when one comes to make a minute study of the form and external structure of a flea, to try and trace the modifications that must have taken place in the course of descent from the ancestral arthropod. But the relationship of fleas to other insects living at the present day is of more immediate concern. Insects are highly specialized arthropods, and fleas are highly specialized insects. This means that they have become vastly modified from the primitive ancestral type and fitted thereby for a life among certain defined and peculiar surroundings. It will be unnecessary to remind the reader who knows anything of zoology or of botany that all classification is now based on descent. Since naturalists have abandoned a belief in the special creation of the various species of animals now living on the earth, and have conclusively shown that they have arisen by descent and modification from other forms, the problem is to reconstruct a vast genealogical tree. What, then, were the ancestors of the fleas, and to what other insects, in consequence, do they appear to be related? It is probable that the ancestors of the fleas were winged insects, and that the organs of flight were gradually lost as they became useless, when a partially parasitic life was adopted. At one time, entomologists regarded fleas as wingless flies, and placed them in the order Diptera. Certain supposed scaly plates on their bodies were regarded as the atrophied relics of wings. It is, however, more than doubtful whether this view is correct, and all modern entomologists who have given any special study to fleas are agreed that they are sufficiently unlike any other living insects to deserve a place in an order by themselves. To this order, the name Siphonaptera has been given, which means that the insects comprised in it are provided with sucking mouths, and are destitute of wings. Another name for the order is Aphaniptera, but this is gradually falling into disuse. Linnaeus, 1758, only mentions 2 species of flea, the human flea, which he appropriately named Pulex irritans, and the chigoe of hot countries, which he called Pulex penetrans, from the habit which the female has of burrowing under the skin of her victims. At the time of writing, about 460 species of flea have been described and named. But some of the names are doubtless synonymous, and the actual number of separable species that have been discovered is somewhere about 400. The vast majority of these have been described within the last few years, which shows what can be done when attention is turned to any neglected group of animals. There can be no doubt that many undiscovered species still remain, and will now, in due course, be collected, described, and named. The position which should be assigned to the order Siphonaptera in the general scheme of insect classification is a question on which the most learned modern entomologists have disputed with considerable vigor. Some see the nearest relatives among the beetles, others among the flies. The majority, as we shall see later on, would place them near the Diptera, but since no convincing arguments have been produced on either side, it may be wisest to regard the question as still at present unsolved. Fleas belong to one of the groups of insects which go through a complete metamorphosis. Their life history consequently falls into 4 divisions: egg, larva, pupa, and imago. If the climate permits, the female flea lays her eggs all the year round, and from 1 to 5 are dropped at a time. Unlike those of many other parasites, they are never attached to the hairs of the hosts, but appear to be deposited indiscriminately on the floors of houses or in the nests and sleeping places of their hosts. The eggs generally hatch in a few days, and a minute, white, worm-like larva emerges. The larvae of some, and possibly of all, fleas are provided with a wonderful adaptation in the shape of an egg breaker, or hatching spine. This is a thin plate, like the edge of a knife, where the point of the head comes in contact with the shell. The movements of the prisoner make a slight split in the eggshell, which then bursts asunder. This organ has vanished in later larval life, and it is probably lost after the first molt. The larva is legless and has 13 segments. It grows rapidly, and as it grows, molts its skin several times. It is provided with mouth parts adapted for biting, and eats any decaying organic refuse. The larvae may be reared on the sweepings of an ordinary room, or the dirty scurf which collects at the bottom of old birds' nests. It is hardly necessary to add that the mother takes no interest whatever in the larvae, and that the belief that she feeds them on dried blood is not based on any sound foundations. The larval stage lasts some days, and the animal spins a small cocoon before pupating. In the course of a few more days, the time probably depending on the weather, the perfect flea emerges. The larvae generally live in places where the perfect insects will have an opportunity of finding a host as soon as they leave the pupal envelope. The nests of their hosts where the young are being reared are always favorite places. It seems possible that the comparative immunity from fleas which hoofed mammals or ungulates enjoy may be due to the fact that the young beast follows its mother from the time of birth, instead of passing its early life helpless in a nest. Observations made on the development of the dog flea, Tenocephalus canis, in India, show that eggs laid on October 17 hatched on October 19. The larva spun its cocoon on October 25, and the mature flea emerged on November 2. In northern Europe, the human flea takes about 4 weeks in summer and 6 weeks in winter to pass through its metamorphosis. Unlike many parasitic insects, fleas do not constantly pass their time upon the bodies of their victims. The greater part of their life is probably spent on the ground, in the house or nest of the mammal or bird which serves them with blood. In this respect, there is considerable difference in the habits of different species of flea. Some attach themselves to an animal and actually burrow into the skin. These are the most parasitic species. Some only come to feed and leave to lay their eggs. Many probably do not suck blood more than once in their lives. An animal which harbors fleas and which nourishes the adult insect with blood is called a host. No fleas are more than what is called temporary parasites, which means that they pass but a portion of their lives on their hosts and frequently take occasion to hop on and off. All fleas, apparently, go from host to host. The labors of diligent collectors have proved that the great majority of mammals and birds have fleas. As a general rule, it is true to say that certain species of flea are associated with certain species of host. Thus, man is the true host of Pulex irritans. The cat family are the true hosts of the cat flea, Tenocephalus felis, and the dog family are the true hosts of the dog flea, Tenocephalus canis. But the human flea is sometimes found on cats and dogs, and cat and dog fleas occasionally bite human beings. And cat fleas are found on dogs, and dog fleas are found on cats. All fleas, so far as we know, may occasionally pass from one species of host to another, but they do not, for the most part, seem to flourish in unaccustomed quarters. Some fleas are more catholic in their tastes than others. Some seem to be very strictly confined to one host, and even when starving, only suck strange blood under protest. There is a species of flea that has only, except by accident, been found on the long-tailed field mouse, and another that has only been found on the hedgehog. Other fleas are commonly found on two absolutely distinct animals. A good instance of this is the human flea, which, at all events in certain parts of England, is a regular parasite of the badger. As distinguished from true or natural hosts, one must separate what may be termed casual or accidental hosts. All animals which come in contact with one another, or which live in close proximity, may exchange fleas. So even bird fleas may be collected from mammals, and typically mammalian fleas from birds. In this fashion, puzzles may arise which tax the ingenuity of the collector to solve. Bird fleas are sometimes found on bats, and this may be obviously attributed to the bats having inhabited a hole which was tenanted by starlings, or an old loft infested with the fleas of pigeons. All beasts of prey are sometimes found to harbor the fleas of animals they have devoured. Rabbits' fleas are found on wild cats. Hedgehogs' fleas on foxes. Mice fleas on weasels, and fleas characteristic of small birds on stoats. So also in the case of mice, rats, and voles, with holes and runs in the same hedgerow. The parasites usually peculiar to one are not uncommonly found on the others. It is sometimes difficult to determine the true host of a flea. Much more puzzling to explain are the reasons which confine a flea to a certain host, and which cause closely allied hosts to have different fleas. The fleas from the house martin and the sand martin are quite different. Those from the domestic fowl and the domestic pigeon are distinct species. The causes which have affected the evolution of the various forms of flea are too obscure to enable anyone at the present day to offer any satisfactory explanation. Speaking generally, the fleas found on birds have points in common, and they probably form a natural group to themselves. What may be called true bird fleas have been collected from almost all European birds. An unwieldy genus, Ceratophilus, comprises many species of different flea. Some species are very abundant and infest the nests of many different birds. Others are extremely rare. One of these rarities, C. vagabundus, is found in the nests of puffins and other seabirds. Another has been collected on Antarctic petrels. Penguins have a special genus of flea to themselves. A specimen, unique at one time, Ceratophilus borealis, in Mr. N. C. Rothschild's collection, was obtained from the gannet. It has now been found on rock pipits in the Shetland Islands. Two very rare fleas, C. fareni and C. rothschildi, are found in the nests of house martins. Yet the nests of these birds are infested with common species besides. A plague flea, Xenopsilla, has been found on an African swift. 46 different species of flea have been found in the British Islands, but many of these are extremely scarce. We know too little about the geographical distribution of fleas to lay down many accurate generalities. When a great deal more material has been collected and studied, it may be possible to show that certain groups are associated with certain regions of the earth or certain orders of animals. To some extent, this is already seen to be the case. The fleas indigenous to the New World are distantly related to those of the Old World. Broadly speaking, the geographical distribution of the parasite must follow that of the host. But sometimes the parasite is impatient of cold and cannot follow the host out of the tropics. The chigos and their allies are fleas of hot countries. Different kinds of bats are found from the tropics to the Arctic Circle, but the same bat fleas are not found everywhere. When a flea has a cosmopolitan range, it is probable that it has traveled over the world in company with its host. Monkeys have no fleas. This is an assertion that is commonly received with surprise and incredulity. Occasionally, a gorilla or a chimpanzee may get a chigo in its toe, and monkeys in zoological gardens or menageries are possibly exposed to the danger of catching an occasional human flea from the people who crowd round their cages. These are remote contingencies which may happen to anyone. Healthy wild monkeys are much too clean and active to harbor fleas. When they search one another's fur in a fashion that must be familiar to most persons, they are clearing their coats of particles of scurf or of similar scraps of dirt, and not of fleas. So speaking generally, it may be said that no fleas have been found truly parasitic on monkeys. Bats have fleas, but not in great abundance. All bat fleas are rare on their hosts, and extremely difficult to find and collect. The same species are not found on fruit bats and the ordinary smaller insect-eating bats. The geographical distribution of some bat fleas is puzzling. For instance, one species, Ischnopsilla unipectinata, is found on the greater horseshoe bat in Europe, but it is apparently not found on the same bat in the British Islands. In Somaliland and in India, it is found on other bats. With certain exceptions, ungulates are remarkably free from fleas. This great order of mammals includes a variety of hoofed animals: oxen, sheep, goats, deer, pigs, camels, giraffes, and antelopes. The only true fleas found on these are two species of the genus Vermipsilla, which resemble the chigos insofar that the pregnant females burrow into the host and expand there. One species has been found on camels and horses in Transcaucasia, another on roe deer in northern China. The female of the last is often found ensconced on the inside of the nostrils of the deer. Of course, chigos may attack domestic ungulates of all kinds, but no other members of the family Pulicidae, or typical fleas, except those two above mentioned, have been found on hoofed mammals. Insectivora such as moles, shrews, and hedgehogs are the hosts of a great variety of species. The same thing may be said of the rodents, which include porcupines, squirrels, rats, mice, and a vast number of other small mammals whose geographical distribution includes almost the whole of the habitable globe. Probably more different species of fleas have been collected from insectivora and rodents than from all the other orders of mammals grouped together. The carnivora, excluding the Pinnipedia, or seals, sea lions, and walruses, harbor numerous species. Among the Edentata, a very remarkable and highly specialized genus of fleas is parasitic on armadillos in South America. This genus, Malacopsula, consists of 2 species only, which are confined to South America and are found on the armadillos and on carnivorous animals which probably have preyed on them. The thorax of these fleas is much reduced and very small in size. Their piercing organs are slender and weak, but they possess enormous spines on the legs, with which they hold on to their hosts. These 2 South American fleas, M. grossiventris and M. androcli, will be referred to again later as striking examples of fleas with strongly developed legs and weakly constructed mouth parts. The contrary combination of powerful mouths and degenerate legs is also found in other groups of fleas, as will be seen in the chapter on the chigos. The marsupials of Australia and South America have special fleas which were probably associated with this strange order of pouched mammals before they became divided into the American and Australian groups. Fleas have been collected on the spiny anteater, Echidna, which belongs to the lowest order of monotremes or egg-laying mammals. On almost every form of bird, including the most aquatic kinds, fleas of various species have been obtained. Only one instance has been recorded of a flea occurring on a reptile. A female of one of the species of burrowing chigos, Echidnophaga ambulans, from Australia, was collected by Dr. Woodward from the brown snake, Dymenia superciliosa. This reptile, which is well known in Australia, belongs to a subfamily that contains some of the most deadly poisonous snakes and is allied to the cobras. The brown snake is a terrestrial snake, and one must regard the presence of the flea on such a host as a rare and chance occurrence. The snake was captured at Herdman's Lake near Perth in West Australia. The same species of flea has also been obtained from the phalangers, Trichoceros, which live in the tops of the Australian gum trees, from the little terrestrial and nocturnal rat kangaroos, Batongia, and from the banded anteater, Myrmecobius, another Australian marsupial. It is possible that the flea moved from some small mammal which was being devoured by the snake and managed to fix itself between the scaly plates of the reptile. When fleas are hatched in a nest, they have no choice but to attach themselves to the young mammals or birds. But even in that case, they frequently leave their hosts and do not for very long remain stationary. Moreover, when a host dies and becomes cold, the fleas invariably leave their quarters, which explains how it may happen that carnivora get infested with the fleas of their prey. This change of hosts, which is always occurring, makes it impossible to draw conclusions from material collected in zoological gardens, where many animals are herded together. In menageries, too, the normal conditions of breeding are absent. A German naturalist collected 2,036 fleas from theaters, concert halls, ballrooms, schools, and barracks in the Grand Duchy of Baden, and found that more than 50% were dog fleas, Tenocephalus canis. What the proportion may be in other parts of Europe, we have no materials from which to form a judgment. In zoological gardens, cat fleas, C.T. felis, are generally numerous in most of the cages. It is, of course, well known to every zoologist that species are not fixed or constant, and that various forms of mammal or of bird tend to show geographical variations. When a long series of skins are laid out on a table and carefully examined, it is seldom that those from the west of any great region cannot be picked out and distinguished from those obtained in the east. So we also get northern and southern forms of the same species varying slightly. These variations are perceptible in many forms of insects, and zoologists now describe these local races as subspecies and designate them with trinomials. No one, however, knows enough as yet about all the various forms which are assumed by fleas to attempt, except in a few instances, to do so in the case of these animals. The study of Siphonaptera is still quite in its infancy. We know little or nothing of the minute geographical variation of fleas. That there is such a thing can already be seen in a few species. In the meantime, the study of variation must be postponed until collectors have amassed a more plentiful amount of material, and it is best to treat all forms which are to all appearance constantly different as being specifically distinct until more is known about variation. Any classification of fleas that may now be attempted can only be tentative. It will be enough, for present purposes, if the reader will remember that the order Siphonaptera can be divided into 3 groups or families. 1. The chigos and their allies, which are the most parasitic fleas, Sarcopsylidae. 2. The typical fleas to which the majority of species belong, Pulicidae. And 3. The bat fleas, Ceratopsylidae, which have certain peculiarities that will be described in a later chapter. Of the antiquity of fleas, and of the period in geological history when the order made its appearance, little can be said. When it was thought that fleas were confined as parasites to warm-blooded mammals and birds, evolutionists were inclined to say that the parasites could not have appeared before their hosts. The discovery of a flea on a reptile opened the vista of possibly enormous antiquity stretching back to Permian or Carboniferous ages. The fossil record is most meager. If we reject as too doubtful the supposed remains of a flea from the lower Oligocene strata at Aix-en-Provence, only one undoubted fossil has been discovered. Nor does it seem certain that fleas are entirely restricted to preying on vertebrates. Dr. Dampf introduced a number of common bird fleas, Ceratophilus gallinae, of both sexes, to some hairy caterpillars. He observed that several of the fleas buried their heads in the hairy covering of the larvae and remained some time in the attitude of sucking blood. While this was going on, the victims made violent demonstrations of annoyance and discomfort. He also observed that a naked caterpillar was not attacked. Mr. Bowden has also recorded how he found in a seed warehouse some peas that were being eaten by 2 species of Lepidopterous larvae. On bringing these home and keeping them in a jar, he found among them some small larvae which ultimately turned into fleas, probably Pulex irritans. These fleas, being confined without other food, were observed to prey on the Lepidopterous larvae, and to feed freely on their juices. The larvae, which were attacked by fleas, pined and died. The fluid from the stomach of the fleas when they were crushed was transparent and not red like vertebrate blood, which often exudes when a mammalian flea is pinched and cracked open. A French entomologist has also reported that the numerous fleas which swarm in the dwelling houses of Corsica, for want of other nourishment, turn their attention to flies that may be incapable of flight. The only fossil remains of a flea that have so far been discovered are a single insect in a piece of Baltic amber of Oligocene age. Many organic remains have been preserved in this fashion, but this is the first mammalian parasite that has been found. The flea is admirably protected by its semi-transparent surroundings and the most minute details of structure, the arrangement of bristles on the body, and the number of segments to the labial palpi can be discerned. This unique object is in the collection of Professor Klebs. The first point to note is that a flea of this antiquity hardly differs from the existing insects of the present day. It has been referred to an existing genus, Paleopsila, of which there are at least 4 species living. 3 of these are parasites of the mole, and the 4th is found on shrews. There is good reason to suppose that the host of the fossil was some insectivorous mammal. The early specialization of fleas is strikingly illustrated. This insect is already adapted for life on some warm-blooded animal. It has a thoracic comb, and its mouth parts are in all respects like those of a modern flea. It belongs to a genus which is still commonly distributed over Europe. When we consider how remote are the chances that a mammalian flea should first get embedded in amber and should subsequently be detected and described by a naturalist, we may well understand that the owner of the fossil asked, though without success, 1,200 pounds for it. The ordinary person regards fleas as a subject for humor of an obvious and familiar kind. The utilitarian despises a man who can cheerfully spend his time in collecting fleas. Yet it seems probable that a study of their forms and habits may be of immediate benefit to the human race. The discovery that fleas are connected with the spread of plague is an instance of apparently unprofitable scientific labor proving of direct advantage to mankind. An accurate knowledge of the structure and habits of fleas is now seen to be of importance to all who are engaged in fighting one of the most dreaded infectious diseases. When plague breaks out, men of science now at once turn their attention to the fleas. This is likely to prove more directly efficacious than the medieval custom of marking the house with a red cross and inscribing the legend, "God have mercy on us." End of Chapter 1. Chapter 2 of The Flea. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. The Flea by Harold Russell. The external structure of a flea. In comparing the structure of a flea with that of a man or any other of the higher animals, it is of the utmost importance to understand that the one has an internal and the other an external skeleton. In either case, the skeleton serves as an attachment for the muscles by which the animal moves itself. Everyone is familiar with the external skeleton of a lobster and can see for himself how the muscles are attached. The structure of a flea, though so much smaller, is somewhat similar, except that the skeleton is composed of a horny substance known as chitin instead of being calcified. The chitinous cuticle entirely covers the flea, but it varies in hardness and thickness on different parts of the body. The epidermis, or true skin, lies immediately beneath. On those parts of the body which are to the rear of the head, the chitin forms a series of plates or shields which overlap one another somewhat like the tiles of a house. The segmented structure of a flea is there most clearly seen. This, we may suppose, is an inheritance from the segmented worms. The chitin which forms the external skeleton of a flea is secreted by an outer layer of cells on the insect's body. The deposit being thin at the joints and thick on the plates, which serve for protection, the flea is encased in a suit of flexible armor. It is made of a fairly solid and dense substance, but, owing to the absence of carbonate and phosphate of lime, is much lighter than the familiar external skeleton of the lobster. Chitin is a very peculiar and durable substance which resists boiling in acids or alkalis. It is a structureless substance, in the sense that it does not consist of cells. Though horny in appearance, it is, of course, in no sense true horn like that of the nails, hoofs, claws, and horns of vertebrates. The different species of flea vary considerably in size. Some are smaller than the familiar human flea. Others are much larger. A very large flea, Hystricopsila talpi, is that found on the mole. The largest known flea, Dolichopsilus stylosus, is found on small rodents in the United States, where, as we know, all things are on a bigger scale than in the Old World. It is 7 millimeters long. The color of the horny integument varies from a pale or light yellow to a ruddy or dark brown. It is plentifully sprinkled over with spines, bristles, or hairs, directed backwards so as not to impede progress. The presence or absence, the arrangement on the body, and the size of these serve, along with other features, to distinguish different species. They seem materially to help a flea in those wriggles to escape with which we are all familiar. The bristles are not always the same in size and arrangement in the two sexes of the same species of flea. As a rule, the males are more bristly than the females. These appendages of the flea's integument are called by various writers either spines, bristles, or hairs. There is, however, no real distinction in the structure or nature of the appendages, and it is a question of degree which name is most appropriate. In distinguishing species, very little reliance can be placed on the color of the flea. An insect newly emerged from the pupa is always lighter in color, and the difference between the appearance of an empty stomach and a stomach gorged with red blood is surprising. The general external appearance of a typical flea belonging to the main family Pulicidae is fairly familiar to most persons. This is well seen in the figure of the oriental rat flea. The body is compressed or flattened from side to side, and this is a feature which is extremely rare among insects. It doubtless enables the animal to glide with greater facility through the hairs of its host. Like other insects, a flea is readily divided into a head, a thorax, and an abdomen. The head is rounded on the top and front and shows no obvious trace of segmentation, but what is known of the development of other insects leads one to think that it must properly be regarded as a number of segments closely fused together. On the underside of the front part of the head is a beak or proboscis for piercing and sucking, composed of the mouth parts, whose structure is worthy of minute study. It will be best to examine them in detail in a subsequent chapter. Some fleas have eyes, others have none. The common mouse flea, Leptopsila musculi, is blind. The bat fleas are also destitute of eyes. The nocturnal habits of their hosts would render eyes of little or no use. If eyes are present, they are large and placed on either side of the head. Each is a simple eye, or ocellus. The compound eyes, divided into a great number of hexagonal facets, which are characteristic of many insects, are never found in fleas. Nothing is known about the flea's powers of vision, but there is no reason to suppose that they are at all acute. The eyes are marked with pigment. Ocelli appear to be primitive types of insect eye, which are perhaps an inheritance from a worm-like ancestor. Presumably all the fleas of long ago had eyes, and those that are now blind have lost their organs of sight from disuse. In their simplest condition, the eyes of the lower invertebrates only enable the creature, so far as one can judge, to distinguish light from darkness. Entomologists believe that the power of vision of ocelli is probably confined to very near objects, and that this simple form of eye is more useful in dark places than the compound eyes. There is no reason for believing that fleas can distinguish colors or can discern any object which is more than a few inches away. It is enough for their purpose to perceive from which point light comes upon them and to make all dispatch to escape in the opposite direction. In blind fleas there is often a spine where the eye should be. In one species the spine is rudimentary and there is some black pigment beneath it. It is not impossible that this is the vestige of a once functional eye. In one genus, however, the eye and the spine are both present. Of the fleas belonging to this genus, one species is South American and the other European. The latter, Typhloceras papei, is confined to the long-tailed field mouse. The organs by which fleas keep in touch with the outward world, and with other fleas, are their antennae. All fleas have antennae, but unlike those of a moth, a beetle, or a grasshopper, each fits neatly into a groove at the side of the head, and can be protruded when desired. This is another adaptation to enable the insect to creep swiftly through a forest of hairs. The combs which are found on the heads of many fleas are organs of exceptional interest. They are toothed and horny appendages which are connected with parasitic habits, for somewhat similar combs are found on several unrelated groups of parasitic insects, as for instance on parasitic beetles, Platypsilus, found on the beaver, on insects allied to bedbugs, Polyctenes, found on bats, and on wingless flies, Nycteribia, which infest Egyptian and South American bats. The majority of the Pulicidae have one or more combs with comparatively long teeth. These combs reach their maximum development in the bat fleas, which have no less than 8. Some Australian and South American fleas, Stephanocercus, have a helmet-like comb extending all round their heads. These combs are by some supposed to be of service in holding on to the hairs of the host, and if one may judge from experiments made on live fleas in cotton wool, they are also used in moving forwards through the fur. All the chigos, Sarcopsylidae, have a large, triangular, post-oral process which is more or less curved and probably prevents the flea slipping back as it pushes forward. Bat fleas, Ceratopsylidae, have lobes or flaps placed 2 on each side of the head, which may possibly serve an identical purpose, but whether they do so is not known. These combs may be divided into 3 groups according to the part of the flea's body on which they are found. Those found on the head are called genal combs, and take the form shown in Figure 2. There are also combs found on the thorax. The fossil flea, Paleopsila klebsi, described in the previous chapter, has one of these thoracic combs. A certain number of fleas also have combs on the abdominal segments. There are really 2 types of toothed organs on fleas, to both of which the name of comb may be given. One is composed of a sheet of chitin with a number of slits and teeth, and resembles a true comb. The other consists merely of a number of highly chitinized bristles arranged in a row. They probably both serve the same function. Apart from their use as organs to assist movement onwards, they may also serve as hair-tight joints and protect the flea from the inconvenience of getting the tips of the host's fur into the joints of its horny armor. The size of the head compared to the thorax and abdomen varies considerably in different species. Some fleas have what may be called by comparison large heads, and others very small ones. A small head is never found in a flea with powerful mouth parts. The head being the bearer of piercing and sucking organs, which require strong muscles, there must be room not only for the organs but for their extensors and retractors. There are normally 3 rows of bristles on a flea's head which divide the head into 4 sections. It is possible that these correspond to the 4 segments of the ancestral insect, which are now fused together. The head of a flea is closely applied by the whole of its back surface to the body, and that slender and conspicuous neck, which is characteristic of the Diptera, or flies, is not to be found in any fleas. For this reason, a flea cannot turn its head in any direction without at the same time following it round with its body. The thorax of a flea consists of 3 segments, called respectively the prothorax, mesothorax, and metathorax. The chitinous external skeleton which covers each of these 3 segments is primarily a hoop, but each hoop is further subdivided into a number of complicated plates. Attached to the thorax are the 3 pairs of legs which are characteristic of all adult insects. The hind pair are very much the strongest. They are the organs of hopping. It has often been pointed out that if men had the leaping powers of some fleas, they would bound with ease backwards and forwards over the cross on the top of St. Paul's Cathedral. Each leg consists of 4 segments beautifully articulated and plentifully supplied with bristles. At the end comes the foot, with 5 very short segments. The last segment is provided with a pair of more or less formidable claws. Fleas use their legs for leaping, for running, and for clinging to their hosts. They also use their mouth parts for the last purpose, and it is worthy of note, as we shall see later on, that in those fleas in which the mouth parts are shortest and weakest, the legs are most liberally supplied with bristles and possess the stoutest claws. The legs of a flea are unique in the insect world, owing to the enormous development of the segment nearest the body called the coxa. Most leaping insects rely for their activity on the muscles of the lower joints. In a grasshopper, it is the third joint from the body, femur, which is so immensely enlarged. The 3 pairs of legs are each attached to a different thoracic segment. When fleas walk, they are, so to speak, plantigrades, walking on the sole of the foot, and all the tarsal or foot joints are applied to the surface of the ground. The claws serve as grips, so as to make the most of any unevenness, and thus the insect drags itself along with surprising rapidity when it moves through the hairy coat of a mammal. But on an open surface, fleas are not really rapid movers compared with many other insects. The 2 claws on the end of each ultimate foot segment are freely movable and are, in fact, highly modified bristles, or setae. In all fleas, one of the plates of the metathorax, or hindmost thoracic segment, called the epimeron, is large and prolonged towards the rear. It invariably bears a stigma. The epimeron is placed laterally to the first abdominal tergite. The older naturalists jumped to the conclusion that this was the remains of a wing. The best judges have, however, formed a decided opinion that no trace of the relic of a flying organ can be detected on the thorax of a flea. Hymans, a German entomologist, has also failed to detect any sign in dissections which he has made of the larvae and the pupae. The epimeron is in fact neither a scale nor a wing, but a portion of the thorax present in all insects. It is of no special service to the flea except as a portion of the thoracic armature which covers the body. The larva of a flea has no legs. The adult insect has 6. A study of other embryo insects shows that the ancestors of insects had many legs. It is an interesting problem why insects lost the legs on their abdomens, why legs should now invariably be restricted to the thorax, and why there should never be more than 3 pairs. In the earliest known insects which lived on the earth, before winged forms were evolved, the number of legs was already 6. But our knowledge of fleas is too small to attempt at present to trace their exact line of ancestral descent. The abdomen of a flea consists of 10 segments. The horny plates which cover the dorsal side are called tergites, those on the ventral side sternites. In fleas, as in all holometabolous insects, that is, those which pass through a complete metamorphosis, the sternite of the first abdominal segment is suppressed and has completely disappeared. The tergite which covers the dorsal part of the first abdominal segment nearest to the thorax is, however, always present. The ultimate segments of the male and female flea are modified for reproductive purposes, and of these segments more must be said later. Having now given a rough outline of the external skeleton of a flea, it only remains to say something about the muscular system. Attached to the inside of the chitinous armature are an enormous number of muscles, whitish and almost transparent. They act as extensors, retractors, flexors, elevators, and depressors. The joints and hinges of the skeleton allow of considerable but not perfect freedom. The muscles of locomotion are partly in the thorax and partly in the several joints of the legs. Our knowledge of the muscular system of fleas is very imperfect, but as in other insects, the general arrangement of the muscles is based on the segmented structure of the body. For the reader who can accurately picture to himself the external structure of a flea and of the typical insects belonging to other orders, a few words may be said on the probable ancestry of fleas and their relationship to other living insects. This vexed and much debated question is still, as the older naturalists would have said, tremendum mysterium. Very little light has yet been thrown upon it, and the most divergent views have been expressed by learned and competent entomologists. A historic survey of the various opinions that have been held since the days of Linnaeus would fill many pages, but a short summary of the different orders to which fleas have been referred by different zoologists will suffice. The older authors, Linnaeus, Geoffroy, Cuvier, and Dumeril, and Gervais, placed them among the Aptera because they were wingless. Kircher regarded them as Orthoptera, an order which includes grasshoppers and crickets, but he has had few followers. By Fabricius and by Illiger, they were treated as Hemiptera, or bugs. Lemire, a Belgian, has recently expressed a decided view that fleas are really a family of Coleoptera, or beetles. Those who have held the once orthodox opinion that they belonged to the Diptera, or flies, are Rosel, Oken, Strauss-Durkheim, Burmeister, Newman, Walker, von Siebold, and Wagner. The structure of an adult flea, however, differs from that of an adult fly in the following noteworthy respects. The mouth parts are differently constructed. The head of the flea is closely joined to its thorax. The three divisions of the thorax are not joined and fused. The flea is wingless. The eyes of fleas are simple ocelli, and there are differences of lesser importance in the stigmata, which give access to the tracheal system by which all insects breathe. The number of those who have regarded fleas as belonging to a distinct order of insects is considerable. They are Lamarck, de Geer, Latreille, Kirby and Spence, Maclay, Leach, Dugas, Boucher, Vanderhaven, Westwood, Landois, Brouwer, Kreplin, and Taschenberg. Modern opinion is all but unanimous on this point. There remains, however, a second question. Even if it be agreed that there must be a distinct order for Suctoria, Aphaniptera, Siphonaptera, or fleas, where ought that order to be placed? In which other order of insects must we look for the nearest relations of fleas? For a time after the acceptance of the fact that insect forms have been evolved and not separately created, the ancestors of fleas were searched for among some species of fly. Then Kreplin rejected the view that flies were as closely related to fleas as most entomologists thought, and his followers could only find points of difference and no points of resemblance. Dahl, 1899, a German, then took up the cudgels for the fly theory. Dahl pointed out the resemblance between fleas and a group of flies called Phoridae, also parasitic on warm-blooded animals. During the ensuing years, the debate was resumed afresh with much liveliness and sometimes with a little acrimony. The fleas were placed by Maclay and by Balbiani between the Diptera and Hemiptera, by Leach between the Hemiptera and Lepidoptera, by Dugas between the Hymenoptera and Diptera, by Brouwer between the Diptera and Coleoptera. Handlersch thinks that fleas have no connection at all with beetles, and Gross can find no signs of relationship with either Coleoptera or Diptera. Embryology and the study of larval forms have thrown so much light on the ancestry of many animals that it was hoped that a microscopic examination of the larvae of fleas in various stages of development would produce some facts of importance. In this hope, entomologists have, to a great extent, been disappointed. There seems to be much similarity between the embryos of beetles, moths, flies, wasps, and fleas. Those who have dwelt on the likeness of the larval flea to the maggot of a fly seem to forget that the resemblance to an embryo beetle is nearly as strong. The young larva of the flea is very transparent, and the digestive canal, heart, and nervous system are easily recognized. The eggshell breaker is an interesting example of the development of a temporary larval structure, and it is the only known instance of such a structure in an insect. There are no traces of eyes. The antennae are three-jointed. They are rather long and slender, being about 1/3 as long as the head. The head is well developed, and the larva has no feet. The biting mandibles are broad and triangular. Compared with those of other larvae, they are said to be more like the mandibles of Coleopterus than of Dipterus larvae. The maxillae, or second pair of jaws, are somewhat reduced and rudimentary. The absence of eyes and of legs are points of similarity between the larvae of fleas and flies. The maggot of a fly has also 2 pairs of jaws and a pair of antennae. At the tail end of the larval flea's abdomen are 2 small projections called caudal stylets. They are strong, recurved, chitinous structures which prop up the body of the larva when it creeps and wriggles. There are similar props in the larvae of certain beetles, and no exactly similar organs are known in Dipterus larvae, but caudal stylets are of small taxonomic importance. In one respect, the mature flea is certainly nearer to a beetle than to a fly. The 3 joints of the thorax are free as in a beetle and not fused as in a fly. But when one studies the mouth parts, the true view seems to be that the mouth parts of a flea are equally unlike those of a fly and those of a beetle. Such being the present state of our knowledge, one must wait for fresh light to be thrown on the matter by further researches. It seems unlikely that the immediate future will produce a solution of the problem. End of Chapter 2.
Chapter 3 of The Flea. This is a LibriVox recording. All LibriVox recordings are in the public domain. For further information or to volunteer, please visit LibriVox.org. The Flea by Harold Russell. Chapter 3. The Mouth Parts and Sense Organs. When the outward anatomy of a flea was described, in an earlier chapter, the mouth parts, which form a sort of beak or proboscis under the head, were mentioned. These most interesting parts of the insect must now be dealt with. The reader probably knows that some insects have mouths for sucking fluids and other mouths for biting solids. A moth or a fly cannot masticate solids, whilst a beetle or a cricket has effective biting jaws. The first naturalist who studied the mouth parts of a flea with such microscopes as were then available was Leeuwenhoek. He was a Dutchman who worked at the end of the 17th century, and the minute accuracy of whose observations still often fills modern naturalists with wonder. Microscopic work was then in its early days, but Leeuwenhoek clearly made out the 2 serrated lancets, which are called the mandibles. His microscopical observations on the structure of the proboscis of a flea were published in the Transactions of the Royal Society in 1706. The mouth parts of fleas are differently constructed from those of all other insects. Around the orifice of the mouth are a number of appendages, which form a complicated apparatus for piercing and sucking. Their construction and use cannot be described without employing some technical terms. When the names of the parts have been mastered, a diagram will make their relative positions clear. It may be necessary, first, to remind the reader who is not an entomologist that the real mouth of an insect is the entrance to the alimentary canal, and that the appendages of the mouth, which act like jaws for masticating or like tubes for sucking, are really modified limbs. In fleas, the mouth is suctoral. But before sucking up the blood, the flea must first pierce the skin of its host. The paired mouth parts, then, are modified limbs, which correspond to those appendages on the thorax of an insect which we call the 3 pairs of legs. The primitive insect, of which fleas and all other insects are descendants, was, it is supposed, composed of a succession of segments each bearing a pair of jointed appendages. Insects of the present day never have more than 6 legs, but the foremost pairs of appendages have been bent round, reduced in size, and altered in shape so as to serve as mouthparts. Now the mouthparts of the flea, for which only technical names exist, are the maxillae and maxillary palpi, the labium and labial palpi, the mandibles, and the labrum. The labrum is considered by some authorities to be the hypothylax. It will be best to deal with each of these in turn and then to explain how they act in combination. The maxillae, these are a pair of horny or chitomous triangular plates, one on either side of the flea's face. They are placed some distance away from the orifice on the mouth and to the right and left of it. They do not serve for piercing or sucking, and appear to have no active functions unless they serve to separate the hairs of the host and enable the flea to reach the bare skin. In the majority of bat fleas, Ceratopsidae, the maxillae are dumbbell-shaped, but in all other fleas they are more or less triangular. From the forepart of each springs a palpus. Like other highly chitomous parts of a flea, the maxillae are usually dark in color. The maxillary palpi. These are jointed hairy feelers which project forwards and were mistaken by the older naturalists for antennae. They spring from the base of each of the maxillae, where these latter organs are joined to the head of the flea. The palpi are sense organs, as the number of sensitive hairs on their surface indicates. The maxillary palpi of fleas are always composed of 4 segments. The labium and the labial palpi. These form together what is called the rostrum of the flea. The labium is a single organ which projects beneath the aperture of the mouth. It may be described as the lower lip of the flea. At its end it divides into 2 comparatively long branches. These are the labial palpi. The actual piercing organs, which will be described below, are the mandibles and labrum. They are not so conspicuous as the rostrum, which protects them. When the piercing organs are at rest, they are partly retracted. The external portion is encased in the tubular rostrum. The tube is formed by the 2 labial palpi, which are situated at the apex of the short, non-divided labium. The number of segments composing each labial palpus in fleas varies. So far as we know, from 2 to 17. In most fleas, however, the labial palpus consists of 5 segments. This appears to have been the original state of things in the ancestral flea. The palpus with more and the palpus with less segments being derived from the normal 5-jointed one. The rostrum of a flea is not a piercing organ like that of a fly and a bug. The 2 labial palpi separate and lie flat, right and left, on the skin when the true piercing organ is driven into the host. The labial palpi therefore require to be flexible, and this is attained by increasing the number of segments or by reducing the amount of chitomization or horniness. We shall find in the chigos and other allies a rostrum which is pale, weak, soft, and scarcely horny. Among other fleas, where the rostrum is prolonged and strongly chitomized, we shall find greater segmentation. The small bristles at the extreme tip of the rostrum seem to be sensory organs. They are like those at the apex of the maxillary palpus. When a hungry flea is put on one's arm, it appears to test the skin with these bristles before it ventures to make a puncture. The mandibles. These are a pair of sharp lancets with serrated edges. They make the puncture and are interlocked with the labrum to form a sucking tube. The labrum. This is the central portion of the mouth parts, and is in fact a prolongation of the upper lip of the flea. It is a hard, sharp, awl-like instrument, in shape like a horny trough. Its edges are more or less toothed. Its apex is pointed and it is as long as the mandibles. The general appearance and the relative position of the mouthparts are shown in Figure 4. Bearing in mind, then, that the piercing organs are the labrum and the two mandibles, and that the rostrum, composed of the labium and labial palpi, is merely a sheath, it is easy to form a clear picture of a flea feeding. Anyone who is bold enough to place a hungry flea on the bare skin of the arm can readily observe through a powerful lens what happens. When the flea has chosen a spot to pierce the skin, the rostrum, with the mandibles and the long upper lip or labrum inside it, is moved a little forward. The flea then lifts its abdomen upwards and presses the piercing organs down into the skin. In doing this, it uses its own weight and the strength of the foremost and middle pairs of legs. The hind pair of legs are lifted up into the air. The head can soon be seen coming nearer the skin. The rostrum then divides in the middle. The labial palpi are forced apart as the mandibles and labrum penetrate into the victim's flesh. Finally, they are driven entirely asunder and lie flat on the skin of the host, one to the right and the other to the left. The flea then satisfies its hunger. A stream of blood is sucked up, and when the meal is over, there is a forcible action of the legs, and the mandible and upper lip are withdrawn with a jerk. Numerous observers have remarked on the habit possessed by fleas of discharging the contents of their intestines whilst actually engaged in sucking. In many cases, a drop of bright red blood is squirted from the rectum during the operation of feeding, and this appears to be a common practice among blood-sucking insects. Its bearing on the feeding operation of the flea has not been discovered, but its possible consequence in transmitting disease from host to host will be seen in subsequent chapters on fleas and the transmission of plague. It is said that the nervous systems and brains of fleas are not so highly developed as those of many other insects, such, for instance, as ants, bees, and other hymenoptera. Having drawn attention to the distinction between the external skeleton of a flea and the internal skeleton of a vertebrate, one may with profit do the same in the case of their nervous systems. In both cases, the nervous system senses to convey sensation from the sense organs and movements to the muscles. In the vertebrate, as the reader doubtlessly knows, there is a brain, a nervous cord running from it down the backbone, and a number of nerves issuing from the spinal cord and from the brain in various directions. Here, the main nervous system runs down the back of the animal. In a flea, or other insect, the nervous system consists of a chain of ganglia connected by a nervous cord. A ganglion is a nerve center and, in a sense, each is a brain which may be likened to the one brain of a vertebrate. We have in the cord of ganglia a series of brains, as it were, running from the head down to the extremity of the abdomen. Each ganglion is a mass of nerves, from each of which a fiber passes off to unite with the other fibers and make a nerve. The first ganglion in a flea is placed in the upper part of the head above the gullet. It may be called the brain, since it receives the nerves of the antennae and eyes. In the ancestral insect, we may suppose that there was a pair of ganglia in each segment. Since the head of the flea consists of several fused segments, we may fairly draw the conclusion that the brain is the result of the fusion of several pairs of ganglia. The brain of the insect occupies the same position in the body as the brain of the vertebrate, but the rest of the nervous system lies in the floor of the body under the digestive canal of the flea, whereas in the vertebrate it lies along the back and above the digestive canal. The dorsal spinal cord of the vertebrate is then a ventral nervous cord in a flea. The sensory nerves, which transmit sensations from different sense organs, and the motor nerves, which send stimuli to the muscles, take their origin from other ganglia besides the ganglion above the gullet. In bees and some other insects, it has been shown that the nerves from the palpi and the mouth parts go to the next ganglion, which is beneath the gullet. The same is probably the case with fleas. So when we speak of the brain of a flea, we must remember that it is a relative rather than an absolute claim to that title. A flea has really many brains. In certain blind insects, where the eyes are wanting, parts of the brain are completely atrophied. Whether this is so in the blind species of fleas does not seem to have been investigated. We now pass from the central nervous system to the sense organs of the flea. The chief are the eyes, the antenna, and the pygidium. In regards to the eyes, nothing more needs to be said. The antennae are probably far more important organs to a flea than its eyes, but insomuch as they are at ordinary times concealed in a groove, they are not very conspicuous. The first tolerably accurate plate of a flea by a naturalist we found in Hooke's Micrography, 1664. Robert Hooke, 1635 to 1703, was a somewhat eccentric and irritable man of science who acted as secretary to the Royal Society. His labors were too varied to be effective. He nearly discovered the laws of gravity and also studied fleas. To him belongs the credit of having detected the antennae groove. Just as many of the older naturalists thought that the maxillary palpi were antennae, so others thought that the antennae of a flea were its ears. And when, with the help of their lenses, they saw the antennae erected and protruded from their grooves, they imagined that the insect was cocking its ears and listening after the manner of a horse or ass. But the antennae of fleas are much more to them than ears, though it may be that they are also auditory organs. They are certainly tactile and olfactory organs as well. In outward structure, each antennae consists of two parts which may be called the stalk and the club. The club is divided into a number of segments and is plentifully supplied with hairs. In some species, the cuts which divide the different segments appear to be confined to one side of the club. In others, a sort of central core holds the segments of the club together. The antennae, therefore, are undoubtedly exceedingly complex organs. Such an insect as a flea may well be far more sensitive to movements of the air, vibrations of the earth, smells, light rays, and sound waves than a human being. In their origin, the antennae, like the paired mouth parts, are modified appendages of the fused segments which compose the head of the insect. The fact that there are 4 pairs of appendages on the insect's head, viz. 1. Antennae, 2. Maxillae, 3. Labial palpi, and 4. Mandibles, has been put forward by some entomologists as evidence that the head is formed of 4 primary segments. Antennae apparently enable fleas to find their bearings, to communicate with one another, and to discover the whereabouts of the opposite sex. But it is especially as organs of smell that they play a most important part in the flea's social life. They enable couples to find one another, and, when the sexes come together, the antennae of the male are usually raised and exposed from the groove. Insects generally have some means of cleansing dirt from their antennae. Some make use of their legs, others of their mouth parts. In fleas, there is often a row of short hairs at the hind margins of the groove which may serve as a kind of comb for cleaning these delicate organs of sense. But further observation on this point would be interesting, for no one appears to have seen the comb in actual use. Female fleas are said usually to carry their antennae ensconced in the grooves, whilst the males more frequently protrude theirs. The antennae of the males are generally longer than those of the females. There are certainly noteworthy organs of sense which appear to exist on the upper surface of the flea's head and body. They take the form of small convexities of the body's surface, lentil-shaped and each surrounded at the base by a ring. Somewhat similar sense organs are widely spread through the insect world. As to their function, divergent views are held. Some think that they are for the perception of sounds, some for the perception of light rays, some for the perception of rays of which we are unconscious. Since these organs are placed, at times, in unprominent parts of the body, it seems more probable that they are affected by sound than by light. The preference which fleas show for certain animals, and the repulsion which they manifest on being allowed to suck blood from an unaccustomed host, lead one to believe that they have a sense of taste. This sense in other insects is apparently seated in certain microscopic pits and hairs which form the ends of nerves and are distributed round the mouth. Whether fleas can hear is not, it seems, definitely known. A large number of fleas possess what is called a frontal tubercle. It is a notch in the center of the forehead, but nearer to the mouth than to the antenna. Sometimes the tubercle projects from the groove. This is most marked in the genus of African fleas Lystropisla. The real nature of this organ is unknown. Some regard it as an organ of sense. Its homology is also uncertain. To some it suggests the egg breaker of the larvae, and it is regarded as a relic of the larval stage. To others it suggests an eye, that they regard it as a remnant of an unpaired oculus possessed by the ancestral flea. An exceedingly remarkable organ of sense, which is found in all fleas, is called the pygidium. It is a sensory plate, plentifully supplied with hairs and nerves, and always placed on the back of the 9th abdominal segment. Of all its uses we are still somewhat uncertain, but some observers declare that at the season of love the male flea bestows caresses on the pygidium of the female. In many species, the male flea is sufficiently different in outward appearance from the female to be easily distinguished. The male is usually smaller, and the last segments of the abdomen are so shaped as to give the look of a tail tilted into the air. The frontispiece represents a male flea and shows this well. The internal organs of reproduction, testes and ovaries, in the male and female are placed near the end of the abdomen. The seminal outlet and common oviduct open to the rear of the sensory plate on the 9th segment of the abdomen. The external genital armature of the male flea is exceedingly complicated and quite unlike that of any other insect. When the sexes are united, the usual position is reversed, and the male is beneath the female. It is well known to every entomologist that the hinder segments of insect are often modified for reproductive purposes. In male fleas, it is the 8th and 9th abdominal segments which are altered. In the females, the 8th, and also often a portion of the 7th, has assumed a peculiar shape. The clasping organs of the male flea are portions of the 9th segment and form together a kind of claw reminding one of the pincers of a lobster. It is used by the male flea in the breeding season to detain and hold the female. Every entomologist also knows that the external sexual organs of insects, of both sexes, are of special importance to a systematist or classifying naturalist. They often enable him to recognize the species when other organs do not show sufficiently striking characters. A minute study of the genitalia of fleas is an absolute necessity to the systematic entomologist, the more so as fleas do not present nearly as many or nearly as varied external differences as do the species of most winged insects, where color and pattern of wings are both important. End of Chapter 3. Read by Paul Parkinson, Calgary, Alberta, March 2023. Chapter 4 of The Flea. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. The Flea by Harold Russell. Chapter 4. The Internal Organs of a Flea. A flea, like every other animal, must feed and breathe, which leads to a consideration of the internal organs of digestion and respiration. The digestive canal is a slender tube which connects the mouth and the anus, and which is less convoluted and much straighter than in higher vertebrates. Figure 6 will show the relative positions of the various parts, namely, the mouth, pharynx, gullet, gizzard, stomach, and rectum. Connected with the digestive canal are certain glands and organs of excretion. The alimentary tube itself passes through the middle of the flea's body and is kept in that position partly by muscles and partly by the numerous branching air tubes through which the insect breathes. Above it lies the heart, and beneath it the nervous cord or chain of ganglia. The mouth of a flea, as of any other insect, is merely an orifice which forms the opening into the alimentary canal. Around the orifice are the various mouth parts which convey blood to the mouth, but these, the reader will doubtlessly remember, are modified limbs or appendages of the segments that compose the flea's head. The mouth, then, gives access to the digestive canal. The first part nearest the mouth is the pharynx, which merges gradually into the gullet. Here is placed the pharyngeal pump, which is provided with a sucking apparatus. Muscles attached to the dorsal part of the so-called aspiratory pharynx cause it to expand and contract, owing to the elastic reaction of its walls. The operating muscles which do this are in the head of the flea. When these pharyngeal muscles contract and relax in regular sequence, a rhythmic action of the pharynx itself ensues, and a steady stream of blood is forced or drawn from the mouth stomachwards. In a light-colored flea, under a powerful lens, this action may be watched in the living insect. Behind the pharynx comes the gullet, which leads down to the gizzard. It is perhaps needless to add that this organ, neither in appearance nor in use, bears any resemblance to the gizzard of a bird, which grinds hard food. The food of the adult flea consists solely of liquid blood. The organ called gizzard in the flea, for want of a better name, is, however, remarkable. Its function is not quite certainly known. It is a bulbous expansion in the front of the stomach and situated at the junction of the stomach and the gullet. It contains a multitude of chitinous finger-like processes tapering towards their extremities. From their general arrangement, the complete collection of processes would act as an effective sort of valve and prevent the return of the fluids from the stomach. It seems most probable that this is their function. During the life of the flea, the stomach is constantly churning its contents. Some valvular arrangement between the stomach and the pharynx would seem to be essential. The pharynx is normally collapsed, as the reader may remember, and its walls are drawn apart by muscles attached to its exterior. When the pharynx is full of blood, the muscles relax, the walls collapse like elastic, and the blood is forced into the stomach. In many cases, a flea will feed when the stomach is already tensely full of blood, and some sort of valve is therefore needed to prevent regurgitation into the pharynx when the pharyngeal muscles contract and the walls of the pharynx itself are drawn asunder. This valvular arrangement at the anterior end of the flea's stomach has been minutely studied in connection with recent plague investigations, because there was a theory that fleas carried infection by vomiting the septicemic blood from their stomachs and so transferred the plague bacillus to the puncture which they made in the skin. But an experiment, which has been tried several times, seems to show that the supposed valve is effective. The stomach of a flea which had recently fed was dissected out intact. As long a portion of rectum as possible was left attached at the hinder end. The gullet having been severed, well in front of the valve, pressure was applied with a blunt tool with the object of forcing the blood through the gullet. The hind aperture of the stomach was, at the same time, closed by pinching up the rectum. The result was that, in no instance, was it possible to force blood through the passage which leads into the gullet, yet sufficient pressure was applied to burst the stomach. The stomach of a flea is a pear-shaped sac which occupies an appreciable part of the insect's abdomen. That it is capable of containing a comparatively large amount of blood is apparent from the observation that after a flea has enjoyed a good meal, nearly the whole of the abdomen is seen to be filled with a bright red mass. During the investigation of the part played by fleas in spreading plague, an endeavor was made to measure, as accurately as possible, the average capacity of a rat flea's stomach when filled with blood. Healthy fleas, taken from Bombay rats, were starved for 12 hours, and at the end of that time were fed on healthy animals. The stomach was then dissected out whole and floated in a salt solution. Any adherent organs or muscles were carefully removed. Under these conditions, the stomach can be examined and measured under the microscope. The average capacity of a rat flea's stomach has been approximately estimated to be half a cubic millimeter. The stomach of a flea is therefore, comparatively speaking, very large. The blood remains in the stomach in a partially digested condition. It gradually diminishes in volume, showing clearly that absorption is taking place. At the end of so much of the digestive process as takes place in the flea's stomach, the blood has become reduced to a thick, slimy, dark red mass. This passes down the intestine to the rectum, where it is perhaps further influenced by the secretion of the so-called rectal glands. Finally, the undigested remains pass from the rectum in the form of very minute, round, almost black, tarry drops. The terminal section of the flea's digestive canal is called the rectum. Here are placed the rectal glands, which are 6 in number. Their function seems not to be certainly known. The external opening of the rectum is placed at the extreme end of the flea's body, between the tergite and sternite of the 10th segment. We pass now to a couple of quite distinct appendages of the digestive canal, namely the salivary glands and the urinary tubules. In fleas, the salivary glands are 4 in number. 2 are placed on each side of the anterior end of the flea's stomach. Each is a simple acinous gland embedded in the body and lined with cells which secrete the saliva. The 4 ducts from the pair of glands unite to form 2 ducts, and the 2 ducts thus formed run forward and open into the salivary pump. A spiral chitomous membrane lines the inside of the ducts, keeps them distended, and gives them somewhat the appearance of tracheal tubes. The salivary pump is placed quite in the front part of the insect's head, and is an organ worthy of special notice. It receives the saliva from the glands by means of the 2 salivary ducts which have just been described, and propels it through the exit duct to the pump into the salivary canal in the mandibles. The pump itself is a hollow chitomous organ. Muscles attached to the wall alternately contract and relax, drawing up the salivary secretion and expelling it through the exit duct. The opening of the exit duct is adjusted so as to be opposite to the canals which extend down the mandibles like troughs. It would seem that when the flea is feeding, saliva is pumped into the puncture and blood is pumped out. There is, as it were, an effluent and an affluent stream passing along the mouth parts. The urinary tubules are excretory organs which carry off in solution the waste products of the flea's body. They are sometimes also called Malpighian tubes. This name they received after Malpighi, 1628–94, a famous Italian anatomist, who, 4 years after Harvey's death, saw with his own eyes the capillary circulation of which Harvey had only inferred the existence. He was also the first to detect the urinary tubes of insects. These tubules answer to the kidneys of the higher vertebrate. They vary in number in different insects from 2 to over 100. In fleas there are 4. They are longish, slender, tubular glands which are closed at one end, but at the other open into the rectum. The urinary excretions come from the blood, pass down the tubes into the rectum, and so leave the flea's body by the anus. In insects, the urinary excretion is, generally, only partly liquid. The organs of respiration in a flea consists of a series of tracheae, or air tubes, which open by apertures called stigmata, at the sides of the body. These air tubes branch and form an elaborate system of ramifications. They have a horny lining and are supported by spirally wound, thread-like thickening. In this way, air is conveyed from the external world, and the oxygen, which vital processes require, is conducted to all parts of the insect's body. The blood system of a flea is far less complete than that of the lowest vertebrate. The blood is almost colorless. A large contractile heart drives it into the main blood vessel. There is, however, no closed system of arteries, capillaries, and veins such as the higher animals possess, and the blood circulates in the whole cavity which intervenes between the body wall and the various internal organs. There is little need for an elaborate system of blood vessels, since the internal tissues are supplied with oxygen by the ramifying air tubes. Fleas have more of the air holes called stigmata than any other insects. Each of the 3 segments of the thorax has a pair, as well as the 2nd to the 8th segments of the abdomen. The spiracles or apertures lie free on the outside of the body. In beetles and other insects which run through dusty places, they are lodged in the thin membrane between the segments. The heart of a flea is a very delicate pulsating tube which lies along the back, above the digestive canal and immediately beneath the integument. One may attribute some of the extraordinary strength and vital energy of a flea to the fact that, by the blood system and the air system, the tissues of the body are kept richly supplied with oxygen. The blood of a flea is a thin fluid and, of course, without red corpuscles. The blood that is shed when a flea is crushed comes from the stomach and not from the blood vessels of the insect. The internal organs of fleas cannot be studied without dissection under a microscope. Dissection is best carried on in a solution of salt and water. Fine needles mounted in pen holders are the most handy implements, but the point of even the finest commercial needle that can be bought is too blunt for fine dissections, and it is necessary to sharpen it. This can be done by the help of a rapidly revolving emery reel, varying the inclination of the needle point to the wheel so as to grind off the angles. The flea to be dissected is put in a drop of salt solution on a slide placed on the stage of the dissecting microscope. In the left hand should be a needle with a blunt conical point, in the right a needle with an oblique point. The antennary groove of the flea should then be transfixed and held firmly by the left-hand needle. The point of the right-hand needle is then inserted under the edge of the 3rd or 4th abdominal segments. The segments can then be peeled off by a skillful dissector, much as we peel off the skin of a shrimp for our tea at the seaside. The internal organs of the flea then float off in the salt solution, and by using 2 very fine pointed needles they can be further separated. It is useful to have one needle ready with a hooked end and another fashioned into a minute knife or scalpel. The most conspicuous of the internal organs will be the stomach and intestine. The salivary glands will be found at the side of the stomach with a certain amount of fat round them. Their extraction is not so difficult as might be supposed. The hooked needle can be used to hook the salivary duct. The most difficult parts to dissect are the organs connected with the mouth and rostrum. It is best to remove the head and transfix it with the left-hand needle, then to scalp the head by removing the dorsal half of the chytomys carapace. A bold plunge with the right-hand needle will sometimes effect what is desired. A pull on the labium will sometimes bring out the pharynx. It must be confessed that successful dissections are often obtained more by good luck than by skillful management. The use of diluted potash solution facilitates the study of chytomys parts by gellifying the muscles. End of Chapter 4. Read by Paul Parkinson, Calgary, Alberta, March 2023.
Chapter 5 of The Flea. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Read by Elsie Selwyn. The Flea by Harold Russell. Chapter 5. The Human Flea and Other Species. The human flea, Pulex irritans, appears to occupy an isolated position. The genus Pulex, which Linnaeus established, has now been reduced until it contains one species only. The human flea belongs to the group with eyes and without combs. In some respects, it is the most specialized of all the Pulecidae. The chigos, Sarcopsylidae, resemble it and are doubtless derived from the Pulecidae. The chief structural character of this interesting insect is the greatly reduced thorax, but it can be distinguished from any other known flea by the fact that the upper segment of the hind leg, hind coxa, bears a number of hairs on the inner surface of the posterior portion. A more noteworthy feature in this flea is the presence, in a large proportion of specimens of both sexes, of a small tooth at the edge of the head. This small tooth is sometimes absent, but when present, both its position and its structure indicate that it corresponds to the fifth tooth in the head comb of the dog flea, Catenocephalus canis. In the hedgehog flea, Catenocephalus erinaceae, the teeth of the combs, both on the head and on the thorax, are small in size and few in number. Occasionally, they almost disappear. The conclusion seems justified that the human flea is descended from an ancestral form with combs. To discuss whether the combs become useless and were lost when the host lost the hairy covering of its body would lead into regions of vague speculation and occupy time unprofitably. The nearest allies of the human flea, which are found on various animals, are all inhabitants of the Old World. The indigenous fleas of America are only distant relatives of Pulex irritans. Our knowledge of the present and former distribution of this species is deplorably meager. The many books of travel published in the early part of the 19th century contain hardly any records of fleas. The human flea is now cosmopolitan. Specimens identical with those from Europe are found almost everywhere, but it may be doubted whether this was the case before the great era of travel and steam began in last century. There is one strange and indeed inexplicable fact in connection with the distribution of this cosmopolitan species of flea. It is absent from the oases of the Sahara and the Hausa countries immediately to the south of the Great Desert. These countries have long been in communication with places where Pulex irritans is known to abound. There is no natural barrier. The habits of the natives would encourage fleas to thrive, and other forms of human vermin are plentiful. There is apparently only one explanation that is forthcoming. It is suggested that the soil and climate in these regions of Africa are, for some reason, unsuited to fleas. In other parts of the Dark Continent, where there are European settlements, the human flea seems to thrive surprisingly well and to attack Europeans and natives, as well as wild and domestic animals. In those parts of Asia where there are European colonies and much intercourse between settlers and Orientals, Pulex irritans is a well-established and thriving parasite. Unfortunately, there is no means of knowing whether this was the case among the native populations before European travelers and traders arrived. Pulex irritans has, however, recently been found on the natives of German New Guinea living some 10,000 feet above sea level and in great isolation. Seaports are everywhere infested with fleas. Another problem on which no light has been thrown concerns the evolution of the human flea. It would be of great interest to know whether the present species has undergone modifications of form since it became a parasite of the human race, whether we inherited the species from our simian ancestors, or whether the flea of one of the lower mammals became parasitic on mankind. In the Old World, this flea is essentially a parasite of man. It occurs only occasionally on other mammals. In America, it certainly appears to occur more frequently on mammals, other than man, than it does in the Old World. Human fleas can propagate in deserted human dwellings. The larvae find nourishment in any refuse that has been left behind, and the adult insect can apparently continue for some time to reproduce itself without a meal of any sort, and certainly without human blood. Travelers in the East and in Africa have described how, on entering huts and deserted villages, they have found their clothing covered with myriads of fleas, sometimes ravenous, and at others weak from long fasting. The human flea is a good deal more select in the choice of a host than some other species. The cat flea, Catenocephalus felis, has been found not only on the cat, but also on the dog, tiger, leopard, goat, horse, rat, hedgehog, kangaroo, deer, guinea pig, rabbit, and on man. Many of these specimens collected in zoological gardens. Although when hungry and confined in a test tube the human flea will readily bite a rat or a guinea pig, it has been found that human fleas kept with no other food supply than rats and guinea pigs soon die off. When large numbers of human fleas were wanted for experiments in Bombay, guinea pigs were used as traps to attract them. On one occasion, 2 guinea pigs placed in a house which had been vacant for some days, and in which fleas must have been short of food, failed to attract any of this species, while a man who entered the house shortly afterwards acted as an admirable trap. Those who have not had an experience of the abundance and voracity of fleas in Oriental countries can hardly believe the numbers of human fleas that may be captured by sending a bare-legged man into a deserted house and then picking the fleas off him. In one house, 31 Pulex irritans were taken on a man's legs in a few minutes. In another house, 84 Pulex irritans, 8 cat fleas, and 1 bird flea were caught. In a third, 150 Pulex irritans and 4 cat fleas were captured in a short time. The piercing organs of the human flea are strong and well developed. This is rare in a flea which, far from having adopted stationary habits, is a very active insect. It has been suggested with some show of probability that the wide and strongly serrated mandibles were acquired after man became the host. The naked skin and rough garment of mankind would render the claws and legs of the flea insufficient to keep the insect in a steady position when feeding. Natural selection would, in due course, strengthen the mouth organs. The division of mankind into different races, many of which are quite as distinct as the various species of some genus among other animals, leads one to expect various races among the fleas which are parasitic on them. If the sand martin and the house martin, the rat and the mouse have distinguishable fleas, one might suppose that the Caucasian and the Hottentot, the Australian native and the Red Indian would follow suit. It may be that further study will show that the human flea now consists of a number of different races. In only one case, however, does a development of this kind in fact appear. Fleas taken off Mexican Indians show slight but fairly constant differences from the true Pulex irritans. The specimens are smaller in size, the rostrum is longer, and the clasper of the male is more pointed. If the Mexican Indians have a special race of human flea, it must have developed after the Indians came to America, or they must have brought it with them when they came. In the latter case, this race of flea may still exist in the country whence these Indians originally came. Apart from this apparently constant race, the individual variation in specimens of the human flea is slight. If a large series of mounted specimens are examined with the microscope, it will be noticed that the bristles or spines on the legs are sometimes more or less numerous. But with this exception, marked varieties such as are frequently found among other insects seem to be rare. Although mankind is the true host of this flea, it has been obtained in various parts of the world on various mammals and occasionally on birds. But in England, and probably in other parts of Europe as well, Pulex irritans is an undoubted parasite of the badger. A good series of the insect has been got from wild badgers, freshly captured near Reading in Berkshire, and Hastings and Sussex. In other parts of the world, it has been obtained from a variety of small carnivora, cats, dogs, foxes, jackals, and polecats. It has also been found on rodents, gerbilis, and on insectivora, Erinaceus. In South Africa, it has been taken off a caracal, and in North America off a lynx. Sandy places such as sea beaches and picnic grounds where humanity congregates for pleasure or business frequently swarm with this species of flea waiting an opportunity to feed. The larvae are bred in the sand and fed on organic refuse. The genus most closely allied to that which contains the human flea consists also of a single species only. It is a large flea, Periodontis riggenbacki, found on porcupines all over Africa and in India. Mankind is occasionally bitten by a variety of other species besides Pulex irritans. In hot countries, the chigo, Dermatophilus penetrans, is a serious and troublesome pest, particularly to bare-footed people. In temperate regions, there are rat fleas, cat fleas, dog fleas, and bird fleas, which occasionally transfer themselves to man and feast on his blood. But, on the whole, hunger and propinquity, rather than free inclination, seems to actuate these fleas, of which man is only the occasional host. There are besides very numerous species which have never under any circumstances been known to bite man. There is no doubt that some persons are more attractive to fleas than others. The reason for this we do not know. It may depend on the tenderness of their skin, the quality and taste of their blood, or their personal smell, or possibly all three combined. The various forms of rat flea, which are important in carrying plague from rodents to the human race, are dealt with later on. Among the commonest fowl fleas which bite men are Keratophilus gallinae and Keratophilus gallinulae. Both species infest the nests of many common passerine birds besides the domestic fowl. A common parasite of the pigeon is Keratophilus columbi, which also bites man. Dog fleas and cat fleas frequently transfer themselves to man. It has been asserted that the flea of the dog and the flea of the cat are indistinguishable. Several great authorities on fleas, such as Dr. Carlo Tirabaschi in Italy and Mr. Carl Baker in the United States, have maintained that the differences between Catenacephalus canis and Catenacephalus felis were unreliable and that they are not distinct species. Mr. Charles Rothschild has, however, shown that the two species are abundantly distinct. The males of these two insects can be readily distinguished from each other by differences exhibited in their respective sexual organs. The females can be distinguished at a glance by the different shape of their respective heads. Figure 7, which shows the head of a female dog flea above and of a female cat flea below, illustrates this. It will be seen that Catenacephalus felis has a much longer and more pointed head than Catenacephalus canis. In the males, the difference in the shape of the head is less strongly marked, but is quite perceptible. There are several minor differences in addition which serve, but less clearly, to distinguish these two insects. The first genal spine, or first tooth in the head comb, is shorter in the dog fleas of both sexes than it is in the cat fleas. The abdominal stigmata appear to be larger in a dog flea than in a cat flea, and there are differences in the bristles which seem to be constant. Both species are perceptibly larger than human fleas, and dog fleas have always afforded good material for dissection. Very few dogs seem to be exempt from fleas, and the little pets which are carried in ladies' arms are often swarming with them. This account of a despised and detested group of insects would be very imperfect if it did not mention those educated or performing fleas which have evoked so much astonishment among people who have watched them. It will be best to say at once that the fleas are not educated, and that the performance can only be attributed to their desire to escape. It is stated that a performing flea may be broken of the habit of jumping by being put in a pill box with glass sides which is made to revolve like a lottery wheel. A short course of this treadmill teaches the flea that the objectionable practice of hopping is useless and exhausting. It is said that the life of performing fleas averages 8 months, which seems surprising. They are fed every few days, and the trainers delight in showing the punctures on their arms where the swarm of pets has been fed. Performing fleas are first of all securely fastened, and this is 9/10 of the secret and the art of education. A very fine silk fibre is put round the body and knotted on the back. The flea may then be cemented to some movable or immovable object. It may pull a coach by being attached to a pole made of a bristle. A little paper object stuck on its back is turned by courtesy an equestrian or a ball dress. The lively imagination of the spectators is of great help. The strength of a flea is wonderful, and on being placed on a sheet of blotting paper so that the hooks of the feet can get a hold, the coach travels at a fine pace. In the intervals of the performance, the coach is turned over, and the performer, with its feet in the air, does not get exhausted with needless struggles. Or else the fleas are fixed head uppermost, with their legs extended horizontally to an upright wire driven into the table. Ladies have fans of tissue paper gummed to their limbs. Gentlemen are in the same way supplied with swords made out of fine segments of wire. When two swordsmen are placed opposite each other and the table is knocked, they move their limbs. The swords then clash by chance, and we have a representation of a duel not much worse than may be seen in provincial or even London melodrama. More wonderful are dancing fleas, for there we have a real representation of a ballroom filled with waltzers. The orchestra of fleas, all securely fixed with cement, is placed above a little musical box. The music proceeds from the box, but the vibrations make the fleas gesticulate violently over their musical instruments. The dancers spin round on the ballroom floor. The couples are fastened by a rigid bar opposite each other so that they cannot touch or part. Each is pointed in an opposite direction and tries to run away. A rotary motion ensues, which to the spectators, if not to the fleas, is very like waltzing. End of Chapter 5. Read by Elsie Selwyn. Section 6 of The Flea. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Read by Elsie Selwyn. The Flea by Harold Russell. Chapter 6. The Chigos and Their Allies. The Chigos and their allies belong to a group of fleas sufficiently remarkable to deserve a somewhat detailed account. The reader may remember that they form a family to which the name of Sarcopsylidae has been given. They are the most completely parasitic of any fleas, and the South American Chigo, Dermatophilus penetrans, enjoys the distinction of being the first foreign flea ever described. This pestilent insect, of which the female has the habit of burrowing into the flesh of the host, soon made itself known to the early travelers in the tropics of America. Oviedo, the Spaniard and historiographer of South America, in his Historia General y Natural de las Indias, 1551, seems to have been the first European author who mentions it. After this, the Chigo is referred to by writers of various nationalities in many works which were published during the 16th, 17th, and 18th centuries. It is an insect which appears under a vast number of different names: Chigo, Chigu, Chego, Chigger, Chick, Jigger, Pico, Sico, Migua, Nigua, Ton, and Tsuchike are synonymous. Catesby, in his Natural History of Carolina, 1743, gives a figure of the insect which is easily recognizable. Linnaeus, in 1758, described the Chigo as Pulex penetrans, and apparently did not know much of its appearance beyond what he learned from Catesby's picture. This species and the human flea were the only two which the great Swedish naturalist distinguished by a name, though under the title Pulex irritans he includes a number of different species, such as the fleas from the dog, cat, rabbit, and fowl. The Chigo remained the only member of the family known to scientific entomologists until the year 1860. An allied insect was then found on a South American parrot. A third member of the family was soon after discovered, and is noteworthy because it was the first species recorded from the Old World. It is now known to infest the domestic fowl in all warm countries where these birds have been introduced by man. A fourth species was collected from a South American bat. Up to the present time, some 14 different species, belonging to 3 very distinct genera, have been described, and there cannot be the slightest doubt that, when collectors in hot countries turn their attention to the matter, a great many other forms of this interesting family of fleas will be found. The Chigos and their allies are of special interest for more than one reason. The females are to a greater or less degree stationary. They fix themselves firmly to their hosts and become veritable parasites. Several of the earlier zoologists believed that the animal was a mite, and it is somewhat remarkable that Oviedo himself should have so promptly detected the relationship of the insect he saw with the fleas. By reason of the parasitic habits of the females, more is known about their appearance in life than in the case of the more active males. On some species, the males remain, for the present, quite unknown, and not very much is recorded about the early life history, eggs, larvae, and pupae of either sex. The parasitic habits of the Chigos and other allied fleas lead one to expect peculiar modifications of form, such as are usually to be observed when an animal passes from an active to a stationary life. These modifications are the more easily understood as the various species are not all stationary to the same degree. It is fairly plain that this family of fleas is a development from the less specialized and less parasitic family Pulicidae. In fact, the gradual development of the organs from a generalized to a more specialized stage is strikingly shown in these insects. To follow this in detail would, however, require a very minute and technical knowledge of their form. The Chigo family is so well characterized that a student of fleas cannot possibly have any doubt whether a flea belongs to this family or not. Yet there is great diversity in general appearance as well as in details of structure. One very peculiar character, namely the enormous swelling of the abdomen in pregnant females, is, moreover, shared with certain other fleas. The most distinguishing character of the family, however, is the rostrum. This organ, which it may be well to remind the reader, consists of the underlip and the labial palpi, sheaths, and the piercing and sucking mouth parts. In the Chigos and their allies, the rostrum is reduced, not in length, but in stiffness and in number of segments. In this group, there are never more than 3 segments to the rostrum, whilst in the main group of fleas, with 1 or 2 exceptions, such as the rabbit flea, Spilopacillus cuniculi, there are never less than 5. There is no indication of a comb on the head, but all the family, without exception, have a large triangular projection, which is more or less curved backwards at each side of the head. These 2 organs doubtless discharge the same functions as the combs of other fleas, and prevent the insect from slipping back as it works its way through the fur or feathers of the host. The thorax of a Chigo is exceedingly short. 2 reasons for this may be suggested. In the first place, the jumping power of these fleas is very small, and the muscles in the thorax are consequently reduced. In the second place, the value to the insect of a contracted thorax is obvious, for the abdomen of a Chigo fixed on or in the skin of an animal does not project so much as it would were the thorax of normal length. The troublesome parasite is, therefore, less likely to be rubbed off by the host. In most fleas, the piercing organs of the mouth, upper lip, and mandibles are directed obliquely downwards. In the Chigos, they are directed obliquely forwards. It has been suggested that this forward movement of the mouth is connected with the stationary life which the females assume. Fleas which fasten themselves permanently to the skin of their host do so in a manner similar to ticks. The mouth parts are in a line with the longitudinal axis of the body. This attitude, so far as we know, is assumed by the females of all the family. The females of one genus, Dermatophilus, actually go head foremost right into the skin of their host. The shape of the head is also beautifully adapted to enable the insect to fix itself firmly in a tick-like posture. The forepart is remarkably obtuse and almost has the appearance of being truncate and abruptly cut off. When the piercing organs have been thrown forward horizontally, it must be a great support to the insect, which is fixed by them, if it can press its head down firmly against the skin of the host. The wider the extent of forehead, which can be pressed against the skin of the host, the less the strain on the upper lip and mandibles, which serve as anchors when the host scratches. All the Chigo family have eyes, but in one recently discovered species, the eye is very small and devoid of pigment. Like those of other fleas, the antennae fit into grooves at the side of the head, and the club, which is the sensitive part of the organ, consists of 8 segments. In a good many fleas, the antennae are different in the 2 sexes, but there is no obvious sexual distinction in this family. The peculiar development of the mouth parts is one of the most singular features in the structure of the Chigos and their relatives. These important organs are modified in a fashion not to be found in any fleas outside the family. Here, as in other fleas, the mandibles are piercing organs which penetrate the skin of the host, the upper lip serving in conjunction as a sucking tube. In ordinary fleas, these organs are retracted when the insect has done its meal. In the present family they remain, in the case of the females, apparently permanently fastened in the skin. The piercing organs are broader and the serrate edges of the mandibles more solid and heavy in this family than in the case of other fleas. The 2 methods by which fleas keep in touch with their hosts have already been alluded to. The 2 main fixing and clinging organs are the mouth and the claws. Weak mouth parts accompany strong legs. We observe, accordingly, 2 lines of development. The Chigos and their allies present an extreme case, for the legs are practically useless for holding on. The bristles and the claws as well are exceedingly thin. In this family the mandibles serve the purpose of claws. The other line of development is best seen in a genus of fleas from South Africa, Malacopsilla, where the piercing organs are short and weak, whilst the claws and bristles of the legs are very strongly developed. The modification found in the rostrum of the chigos has already been referred to, and the explanation of this will now be understood. The rostrum is a sheath on either side of the piercing organs, consisting of an underlip and two labial palpi. When the flea sucks, the labial palpi are pushed asunder as the piercing organs are driven in and lie flat on the skin of the host. In this family, the rostrum is almost white in color and soft instead of being horny or chitinized. Where the rostrum is strongly chitinized or very horny, the flea has to use a certain amount of force to counteract the spring-like action of the labial palpi. It is conceivable that rigid labial palpi would prove inconvenient to fleas which remain permanently attached to their host by their mouth organs. The swollen abdomen of the female chigo is a strange and conspicuous object, which is not, however, found occurring to the same extent in all the members of the family. Having burrowed into the flesh of a man or other mammal, the pregnant female swells and enlarges until she reaches the gigantic proportions of a small pea. Itching and inflammation ensue unless the whole insect be skillfully removed with a needle. To such an extent does the abdomen swell that the segments and the horny plates are driven asunder and the connecting membrane between is exposed to view. In the extreme case of Dermatophilus caecata from South America, the abdomen swells until it completely envelops the head and thorax after the manner shown in Figure 8. Dr. Enderlein found 17 specimens of this species in the skin behind the ears of a rat from Brazil. The belief that the eggs are laid in the flesh of the victim is mistaken. The hind segments of the body and their stigmata are always exposed to the air. Stigmata of the 8th abdominal segment is particularly large. As soon as the eggs have been laid, the body of the mother dies, withers, and falls away from the skin of the host. The fact that several females are often found where one has buried herself led to the notion that these parasites bred in the wound. The truth seems to be that other chigos are attracted to a spot where inflammation has made it easy to burrow. Chigos love warmth and drought. The deserted huts of natives swarm with them if they are dry. It is always said that newcomers are more liable to attack than natives, but the explanation of this seems to be that they do not understand the significance of the slight pricks which are felt when the chigo fixes itself. Once the parasite has got under the skin, no pain is felt unless the wound is inflamed by scratching. The tender flesh under toenails is a favorite spot of attack. The only remedy is a sharp knife and a little antiseptic wash. Pigs and fowls are sometimes killed by chigos, and Indians occasionally are attacked by lockjaw after the parasite has been removed. But this is not directly attributable to the chigo. The eggs are laid one by one. When this operation is completed, the mandibles weaken and the shriveled body of the insect can be rubbed off. But a painful sore may be produced if the parasite is forcibly broken off and the mouth parts are left in the wound. The chigo, Dermatophilus penetrans, is a native originally of South America. It ranges from Mexico to northern Argentina. Sometime after the middle of the 19th century, it was, somehow, carried across the Atlantic and introduced into West Africa. From there it has now spread across the dark continent to the Great Lakes and has even reached Madagascar. Such are the modern facilities of transport which parasites are quick to take advantage of. End of Section 6.
Section 7 of The Flea. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. The Flea by Harold Russell. Fleas and Plague. In order to understand the part played by fleas in the transmission of plague, it is necessary to have some clear elementary knowledge of the nature of that disease. Plague is an infectious fever caused by a specific bacterial organism. Bacillus pestis was first identified in 1894 by Kitasato, a Japanese, and immediately afterwards, but independently, by Yersin. It is an exceedingly minute, short, moderately thick, oval bacillus with rounded ends. It has the most astounding power of rapid multiplication. Nothing is, at present, known of its natural history outside the body of the sufferer, but it can be cultivated. Little is known of its toxic action, but a weak toxin has been got from cultures. The bacillus itself is not of a resistant nature and is easily killed by heat and ordinary germicides. Acids appear to be fatal to it. In ordinary cases, the bacillus is found in buboes. A bubo is nothing more than an inflamed gland. In so-called septicemic cases, it is found in the blood of the animal afflicted by the disease. In pneumonic cases, the bacillus may be found in the sputum of the patient. It is the custom to speak of A. bubonic plague, B. septicemic plague, C. pneumonic plague, as though they were 3 diseases. This is inaccurate, for they are only forms with varying symptoms of one and the same disease caused by the same bacillus. The disease which we call plague is, in truth, really a fight between the afflicted animal and the invaded bacillus. It may be inferred from the fact that bacilli are scarcely ever found in the blood in bubonic cases that the invaders are stopped by the lymphatic glands next to the point of inoculation. In such cases the fight, which is the illness, takes place chiefly in the bubo. In non-bubonic cases, the fight goes on in the blood vessels or in the lungs, as the case may be. Whether the plague is primarily a disease of rats would be difficult to say, but rats and other rodents are very susceptible to it. It has also been transferred to mice, rabbits, guinea pigs, squirrels, pigs, sheep, goat, cattle, and horses. Men and monkeys are equally susceptible. Cats and dogs have been known to die of it, and during the Great Plague of London, many were destroyed under the belief that they were bearers of infection. That plague among human beings was associated with mortality among rats and mice is an observation of great antiquity. The student of the Hebrew Scriptures will remember the book of 1 Samuel, chapter 6, verse 4. Then said they, "What shall be the trespass offering which we shall return to him?" They answered, 5 golden emerods, buboes, and 5 golden mice, rats, according to the number of the lords of the Philistines. For one plague was on you all, and on your lords. Eastern authors of a later date refer in several places to rats, in times of plague, staggering about as though they were drunk. The Mogul emperor, Jen-Hajir, in his diary of the plague at Agra, in 1618, mentions the unusual mortality of the rats. In India it seems long to have been a custom, dictated by experience and caution, to leave houses when rats began to die. In Europe, during the Middle Ages, the mortality of rats when the plague was raging does not seem to have impressed the chroniclers, and during the recent outbreak at Glasgow, in 1900, none was detected. As an illness of mankind, the plague reached Europe from the east. We have no evidence of any outbreak in Europe before the reign of the Emperor Justinian. When it raged for the first time at Constantinople, in A.D. 542, the mortality was enormous. 10,000 persons are said to have died in a day, with all the symptoms of bubonic plague. It spread swiftly through the Roman Empire. In the 14th century, the same disease, under the name of the Black Death, again ravaged Europe. Again the mortality was enormous. Millions perished, little suspecting that fleas could be connected with their fate. Everywhere popular tradition reported the plague as the most highly contagious of all diseases. In the history of science, the plague epidemics in Egypt between 1833 and 1845 are of importance, because the disease was, for the first time, seriously studied by skillful French physicians. Some of the French medical school went so far as to deny contagion altogether. The modern view is that aerial infection may be put aside as almost impossible, except in pneumonic cases, but that plague may be transmitted by any method which inoculates the blood with Bacillus pestis. Our modern knowledge dates from the year 1894, when the plague reached Hong Kong. Its existence as a rat disease was recognized. In the autumn of 1896, when plague broke out in India, the men of science, who made careful observations on the spot, were struck by the fact that infection spread from house to house in a fashion that seemed inexplicable, unless the bacillus was carried by an animal. We pass now from rats to fleas. That fleas may be connected with the spreading of plague was suggested in the year 1897, when Ogata first found bacilli in fleas. He obtained fleas from plague-sick rats. These he crushed and injected the liquid into a couple of mice. One of these died of plague in 3 days. The German Plague Commission in Bombay found plague bacilli in fleas, but for various reasons did not consider that the bite of the flea was the means by which the disease was transmitted. The real credit is due to Simond, a Frenchman, who worked during the Indian epidemics. He took fleas from infected animals and observed in their stomachs bacilli identical with B. pestis. He suggested that the bacillus was carried from rats to men, and he brought forward some evidence tending to show that infected fleas could transmit infection by biting. But Simond was not able to bring forward conclusive proof. He pointed out a line of research to others which has proved exceedingly fruitful. In the same year, 1898, Hankin suggested that some biting insect might be the means of transmission from rats to men. The bacillus of plague has now been identified in ants, bugs, and flies, as well as fleas. It seems likely that any suctorial insect which feeds on a plague-stricken rat will take numbers of the bacilli into its stomach. The points which Simond wished to establish were that plague-stricken rats with fleas are exceedingly infective, that they cease to be infective when they have been deserted by their fleas, and that fleas which infest rats will transfer themselves to man. Since 1905, an elaborate series of observations and experiments have been carried out. Post-mortems have been made of countless rats. Numberless fleas have been collected and dissected. But this summary would be very incomplete if it did not mention the work of Verbitsky, a Russian doctor at Kronstadt, whose labors remained almost unnoticed, although he made his experiments as long ago as 1902 and 1903. His thesis, written in Russian, was not published in any scientific journal, but his ingenious and careful experiments showed that fleas could transmit plague from animal to animal. He found that the commonest flea captured off rats at Kronstadt was Leptocilla musculi, the usual host of which in other places is the mouse. Now this flea does not, except very rarely, bite human beings, and the real significance of the facts discovered was not appreciated. The common rat flea in most parts of Europe is Ceratophilus fasciatus, and in India and subtropical countries, Xenocilla chiapis. This last species has acquired the title of the plague flea, or, more accurately, the oriental rat flea. During the plague investigations in India, many careful experiments have been made, proving, beyond doubt, that the disease may be transferred from rat to rat by the transference of fleas from a septicemic to a healthy animal. It was first shown that when fleas were present, the plague could be transferred from rat to rat, kept in proximity, but carefully screened so as to avoid any possibility of contact. Next, fleas were collected from rats dead or dying of septicemic plague and transferred to healthy rats living in flea-proof cages. More than half of the healthy rats contracted plague. It was shown that if fleas are present, the disease, once started, spreads from animal to animal, and it would seem that the rate of progress was in direct proportion to the number of fleas present. The blood of a plague-infected rat may contain an enormous number of plague bacilli. Although such figures do not convey any very clear idea of numbers, as many as 100 million bacilli have been found in a cubic centimeter of rat's blood. A rat flea, with a stomach of average size, might receive, therefore, as many as 5,000 germs into its stomach, and it is clear that fleas, feeding on a large proportion of plague-infected rats just before death, would be almost certain to imbibe at least some plague bacilli. There is, moreover, good evidence for believing that multiplication of the plague bacilli may take place in the flea's stomach, nor does the blood imbibed by the flea cease to be infective when it passes from the stomach. Both the contents of the rectum and the excrements of fleas taken from plague rats often contain abundant and actively virulent plague bacilli. A number of infected fleas are put into a test tube. The mouth of the tube is covered with a glass slide, and the mouth is turned upside down. The fleas are then seen to run over the slide, and in a short time they deposit an appreciable amount of fecal matter on the surface. This, under the microscope, is seen to be covered with plague bacilli, and a large percentage of guinea pigs who have an emulsion of the fecal matter injected into them contract plague. It is remarkable that, so far as we know at present, the plague bacillus is confined to the flea's alimentary canal. On rare occasions, it is found in the gullet, when fleas have been killed immediately after feeding on septicemic blood, but no plague bacillus has been found in the body cavity or in the salivary glands. In the stomach of the flea, plague bacilli have been found in vast numbers 12 and even 20 days after the insect has imbibed septicemic blood. It is naturally of great practical importance to know how long fleas taken from plague-infected rats remain infective. That is to say, are capable of transmitting the infection to healthy animals. 2 series of careful experiments made during the epidemic plague season in India have shown that fleas could remain infective for as long as 15 days. In a 3rd series of experiments made during the non-epidemic season, it was found that the fleas remained infective for only 7 days. It has been ascertained that both the male and the female oriental rat flea, Xenopsilla chiopis, can transmit plague. We come now to one of the most interesting questions of all, namely, the method by which the rat flea transmits plague to a healthy animal. A variety of suggestions have been made, several of which can be shortly dismissed. It was thought at one time that infection might be conveyed by the animal eating the infected flea, but it is very improbable that this means of infection is of any real importance, even if it may sometimes occur. Experiments in feeding have shown that an animal is unlikely to become infected by swallowing material containing plague bacilli, unless the amount is considerable. Moreover, we know that infected fleas, confined in test tubes, readily convey the disease when allowed to bite an animal. In such cases, the situation of the primary bubo corresponds with the area of skin upon which the fleas are placed. That the transmission of plague is due to the bite of the flea seems abundantly clear. It has also been suggested that the proboscis of the flea acts as a mechanical instrument for the transference of the bacilli. No doubt, the outside surface of the flea's proboscis must become contaminated when it sucks the blood of a plague-stricken rat, but it is difficult to suppose that contamination of the proboscis can explain cases of continued infectivity during which the flea has been feeding regularly upon healthy animals. Next, there is a hypothesis that the salivary glands of the flea become infected and that the bacilli are inoculated along with the saliva. The reader will remember that when a flea sucks, a stream of saliva is pumped down the mandibles into the puncture. But this hypothesis is shattered by the fact that plague bacilli are apparently confined to the alimentary canal of the flea and that they have never been found in the salivary glands. An apparently more probable explanation, that the contents of the stomach, in which, as we know, the bacilli may multiply, are regurgitated and transferred to the wound by the mouth parts, is rendered less credible when we remember that there is a valvular arrangement at the opening of the flea's stomach which seems to make such a thing impossible. Lastly, there remains the only theory on which we have positive evidence. It is the theory that the bacilli contained in the feces of the flea are deposited on the skin and then find their way into the wound made by the piercing organ. They may be helped in this by the rubbing and scratching which follow on the bite of the flea. We know, of course, that plague bacilli are present in abundance in the feces of fleas taken from plague-sick rats, and that such feces are infective to guinea pigs, both by cutaneous and by subcutaneous inoculation. Experiments were made to discover whether the pricks made by fleas were of sufficient size to allow plague bacilli to enter the body, no other damage to the skin being done. Healthy fleas, confined in a test tube, were allowed to feed on a small part of a guinea pig's abdomen, the hair of which had been cropped close without injuring the skin. Immediately afterwards, a few drops of the septicemic blood of a rat which had died of plague, or of a virulent culture of plague bacillus, were lightly spread over the part. Many successful infections were obtained in this way. Similar experiments were made in which the plague culture was first spread on the skin, and afterwards healthy fleas were allowed to feed on the same spot. Successful infections were also obtained by this means. Two facts, then, seem to be demonstrated beyond doubt. First, that the puncture made by a flea will allow the bacillus to gain access to an animal's body and to infect it. Secondly, that there is a possibility of infection by the feces of fleas. As to whether this is the usual process, the highest authorities are not ready to express any opinion. The safest course appears to be kill fleas, but to avoid rubbing them in. Good work was done during the recent outbreak of plague in San Francisco, when the energies of an army of men were directed to controlling and destroying the rat population. Enormous numbers of rats were killed, their breeding places were destroyed, and everything was made as uncomfortable for them as possible. Men of science were at the same time engaged in collecting and examining the fleas from many thousands of rats. The great success of the work confirmed the soundness of the theory on which it was based. The spread of the most terrible of epidemic diseases was controlled and prevented by knowledge. At San Francisco, the fleas of man, rats, mice, dogs, cats, ground squirrels, and gophers were studied. It was found there, as elsewhere, that while each species of flea has its particular host, few are unwilling or unable to attack man and other animals when the host dies. There is good reason to believe that during the last outbreak of plague in Manchuria, the fleas carried the bacillus from the marmots, Arctomys, to man. Plague can be transmitted by the human flea, but it may be doubted whether this often occurs under natural conditions. The rat fleas seem inclined to take more readily to mankind than the human fleas do to rats. Experiments at Bombay seem to show that, though the human flea was able to transmit the plague infection, it does not transmit it as readily as the oriental rat flea. An explanation of this was obtained when it was discovered that Pulex irritans does not live well either on rats or on guinea pigs, which were the subjects of the experiments. A count of the fleas was made each day in a number of experimental cages, in which live human fleas were placed in company with wild Bombay rats. A great number of human fleas were put into a flea-proof cage along with the rat. Each day a census was taken of the fleas still alive. After 24 hours, it was found that little more than 1% of the fleas put in could be recovered, and no fleas were ever found alive after the 5th day. The European rat flea, Ceratophilus fasciatus, seems to be quite as readily able to transmit plague as the oriental insect. How far other fleas are able to transfer infection, we have little or no knowledge. But 27 experiments to transmit plague from animal to animal by means of cat fleas, Tenocephalus felis, were once made, and none of these were successful. The reason for the failure we do not know. If infected fleas are kept in captivity after they have fed on septicemic blood, it is found that, after a while, they are no longer able to convey infection. On being dissected, no bacilli are found in them. A clearing process, therefore, evidently goes on. If a number of fleas be fed on a septicemic rat, and subsequently be kept under observation and nourished on healthy animals, the proportion found to be infected steadily diminishes day by day. It is remarkable that the existence of numerous plague bacilli in the stomach of the flea does not seem materially to affect the insect's life. Fleas, in other words, do not suffer from plague, though they can transmit it. End of section 7.
Chapter 8 of The Flea. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Read by Little Miss Clumsy. The Flea by Harold Russell. Rat fleas and bat fleas. The chief conclusions arrived at as the result of the investigations during the years 1905 to 1909 into the mode of spread of plague in India may be briefly stated in the following fashion. The advisory committee, under whose direction the investigation was carried out, consider that, firstly, in nature plague is spread among rats by the agency of rat fleas. Secondly, bubonic plague is not directly infectious from man to man. Thirdly, in the great majority of cases, during an epidemic of plague, man contracts the disease from plague-infected rats through the agency of plague-infected rat fleas. Fourthly, where there are annual epidemics, they occur during some part of that season when the prevalence of fleas is greatest. That being so, it is manifest that an accurate knowledge of rat fleas, their forms, their habits, and their life history may prove of great importance. 3 species of the genus Mus follow quickly in the wake of civilized man and establish themselves all over the globe. They may all be looked upon as more or less domestic animals. The house mouse, M. musculus, is familiar everywhere. The old black rat, M. rattus, chiefly infests ships and seaports. The brown rats, M. noedicus, is the most aggressive and distinctive. But all 3, by accidental transference from port to port in ocean-going vessels, have become distributed over the world. Their fleas, to a limited extent, have become distributed with them. In connection with the spread of plague, these 3 small rodents are of prime importance, and not less important are the fleas which are parasitic on them. In California, the ground squirrel, Otospermophilus bichei, has been proved to play an important part in plague infection, and a full account of its fleas and of experiments in transferring rat fleas to squirrels and squirrel fleas to rats has been published by American naturalists. In 1903, Dr. Blue, who was in charge of measures for suppressing plague at San Francisco, observed that an epidemic disease was killing the ground squirrels in the country round San Francisco Bay. It was shown somewhat later that the mortality among the squirrels was caused by plague, and there can be little doubt that it was transferred from rats to squirrels. In harvest time, rats migrate to the fields and use the same runs and holes as the squirrels. Under these conditions, a transfer of fleas from rats to ground squirrels is almost certain to ensue. 2 species of flea have been recorded from the Californian ground squirrel, and both are parasites of rats. The chain of evidence is really complete, for those who have made a business of hunting ground squirrels testified to the readiness with which fleas will leave a dead squirrel and bite a human being. In the records of plague in California, there are several cases in which there seems to be very little doubt that the disease resulted from handling plague-infected squirrels. Fleas, being wingless insects, travel with considerable difficulty over the ground, and though their hopping powers are notorious, they are unable to make any long-continued progress in this way. The methods by which they get dispersed are of interest. Some may be carried by the host in its natural wanderings. Rats appear to be constantly picking up and dropping fleas. Sick rats harbor more fleas than others, and therefore more frequently drop them. 100 fleas have been collected off one plague-sick rat, and, as we know, if this rat was moribund, some of these fleas would most likely be infected. It is obvious that a plague-sick rat may travel about, leaving as it wanders a trail of infected fleas behind it. Rats, too, are frequently transported with certain kinds of merchandise and carry their fleas to the most distant part of the globe, travelling with all the speed and luxury which modern steamships afford. Rats will dive into sacks of grain or bran and hide, so that the bag can be loaded as cargo without anyone suspecting the presence of a rat inside. M. rattus and its fleas, from the habits of the host, are especially likely to be transported in this way. Besides, many fleas are now dispersed without their hosts in merchandise of various kinds. They may travel great distances in these days of rapid transport, though adult fleas, without a host to feed on, generally die in about 5 days. But larvae, which eat organic rubbish, and pupae, which do not eat at all, might arrive alive at the end of a journey of well over a month. On arrival, they would seek their true host, or the next best available animal. Not having yet fed and being newly emerged, they might survive as long as a fortnight without a suitable host. Fleas dislike damp breeding places, but dirty carpets, chopped straw, old sacking, paper shavings, and such like rubbish suit them admirably. M. rattus is fond of making nests on grain bags, and in such sacking the larvae of fleas are often found. Where trade is carried on in sacks and gunny bags, this means of disturbing fleas and plague should be kept in mind. Some rat fleas, as we know, will feed on man as well as on rats, but their behavior is rather different when they feed on rats and on man. It has been repeatedly noticed that the fleas were much more readily attracted by the rat than by man. Although the fleas jump onto a man's hand, they take some time to begin to feed. They crawl about and seem to have some doubt where best to begin their sucking operations. Also, it has been observed that the fleas much more readily fall off a man's arm when he moves than they do when a rat moves. It seems that they are able to get a firmer hold on the rat than on the man, and it is of interest to note in this connection the larger claws of the human flea compared with the claws of the rat flea. It has been shown in various parts of India that the number of rat fleas found on rats varies with the seasons. This seasonal variation of rat fleas corresponds in a general way with the plague mortality. During the season when plague is bad, the average number of fleas per rat is above the mean. During the non-epidemic season, it is below the mean. The height of the epidemic corresponds fairly closely with the season of maximum flea prevalence. 19 species of Pulicidae are more or less habitually obtained by collectors on rats and mice, but the great majority of these may be called casual visitors. 6 species of Sarcoscylidae are also occasionally found on rats. These are the burrowing chigos and their allies, which usually attack the head and ears of rats. The species of flea commonly found on rats are 5 in number, and the readiness with which they bite human beings has been carefully studied. 1. Xenocylla cheopis. This is the oriental rat flea first described by Mr. Charles Rothschild from specimens collected in Egypt. The true home of this flea appears to be the Nile Valley, where it may be found in plenty on various hosts. Many of these are desert animals, and the flea shows a preference for rodents. Having been distributed all over the world by rats, it now occurs occasionally in all warm climates. It is the common rat flea of the tropical and subtropical world. In India, it often happens that the whole of the fleas collected from rats prove to be of this species, but it cannot apparently flourish in cold countries. In the warmer temperate zones, such as the Mediterranean and Australian seaports, it occurs in varying proportions according to the time of year. The numbers decline with cold weather. It readily bites man and is more active than any other flea in the transmission of plague. For this reason, it is sometimes spoken of as the plague flea. It is a smaller and a lighter colored insect than the human flea. 2. Ceratophilus fasciatus. This is the common European rat flea. It is the rat flea of the temperate, as opposed to the hot countries of the world. It is commonly found on black and brown rats in the British Islands and the other countries of northern and central Europe. It readily bites man, and there is no reason to suppose that, other conditions being equal, it would not be as efficient an agent in spreading plague as the last species has been shown to be in India. 3. Ceratophilus anisus. This is a closely allied species of rat flea which replaces the last in China and Japan. 4. Leptocilla musculi. This is the mouse flea, and it is as widely distributed over the globe as its host. From mice it frequently moves to rats, and it has been found on them in various parts of Europe, America, Australia, and Japan. It occasionally bites man, but evinces little inclination to do so. 5. Stenophthalmus argytes. This flea is commonly found as a parasite of voles and field mice. When farm rats take to an open life in the fields, they pick up this species from the rustic rodents. In Hertfordshire, Hampshire, and Suffolk, 1/2 of the fleas from rats, collected in farmyards and hedgerows, were found to belong to this species. But whether it is as common on rats all over England is unknown. It appears not to bite man. A closely allied flea, C.T. assimilis, is found in Central Europe on field mice and equally on rats which live under the same conditions. It has not been found in England. The principal occasional parasites of rats are dog fleas, cat fleas, fowl fleas, and human fleas. The proportions in which they and rat fleas are found vary greatly in different parts of the world. For instance, in San Francisco, 9% of the fleas collected from rats have sometimes been found to be human fleas, whilst in Italy as many as 25% have been identified as cat and dog fleas. It must be borne in mind that when new countries are opened up by man, the rats which follow in his rear exterminate numbers of the weakly native small mammals and take on their fleas. A change of habitat may be followed by an exchange of fleas. Some interesting work has been done in testing the appetite of different kinds of flea for human blood. The oriental rat flea, X. cheopis, has been kept alive for 3 weeks on that diet. Other species show repulsion for mankind and refuse to suck. The experiments confirm the popular belief that fleas have a marked preference for certain individuals. When the flea has refused to bite the human arm, it becomes necessary to check the experiment by trying whether the refusal is merely due to want of hunger. For this purpose, a rat must be at hand. It can be secured on a board by 2 bondages, fixed at each end by drawing pins. The rat lies, of course, on its back, with its head comfortably supported by a little pillow of cotton wool. A portion of the rat's abdominal wall is left exposed and shaved. The flea, in an inverted test tube, can then be put on the airless patch of the abdomen and given an opportunity of biting, which it may or may not accept. When fleas are being collected from rats, it has been noticed that the true rat fleas are usually on the hindquarters of the host, whilst the mouse flea prefers the region of the head and neck. As regards the tastes and habits of oriental rat fleas in the matter of food, a long series of experiments may be summarized in this way. 1. When many rat fleas are present, some will attack man, even when a rat is available for their food supply. 2. When the number of rat fleas is small, and when their true host is present, they will not attack man. 3. When rat fleas are starved, they will readily attack all animals, not being particular in the choice of a host. 4. Rat fleas, deprived of their food for from 72 to 96 hours, attack and feed on man more readily than at other times. 5. Rat fleas, even when starved, prefer their true host to man. 6. Rat fleas may be attracted to man, jump on him, but take some time to feed on him. Plague-infected fleas might in this way be carried from one place to another without infecting the man, but they would, when brought near a rat, attack it in preference to man. The fleas found on bats possess certain peculiarities which have led to their being grouped together. They form a family to which the name Ceratopsylidae has been given. They are recognized by 2 flaps, one on each side of the head. What these are and what service, if any, they render to their possessors is unknown. Bat fleas also, as a rule, have maxillae shaped like dumbbells, but in one genus, Thomopsila, found on fruit bats, they are triangular as in other fleas. The maxilla, as the reader may remember, are parts of the insect's mouth, and though placed like jaws on each side of the aperture, they are not used in piercing the skin and sucking blood. They bear feelers called the maxillary palpi. The flea, Thomopsila breviceps, which is found on South African fruit bats and which has triangular maxilla, seems to be a connecting link between this peculiar group of fleas and the main family Polychidae. Bat fleas are commonly well supplied with combs. They usually have them on the abdomen as well as the head, and the maximum number of 8 combs is found in bat fleas. Their structure and life history agree generally with that of other fleas. They breed in hollow trees, caves, ruins, church towers, and lofts where bats hibernate or spend the hours of daylight. The larvae feed on the droppings of the bats, and the mature insect, after emerging from the pupa case, takes the first opportunity that comes of getting on to its host. Bats are seldom found to be much infested with fleas. For this reason, bat fleas are somewhat difficult to obtain, and many of the species that are known are extremely rare. The hosts of bat fleas obviously vary more as to the surroundings which they inhabit than almost any other animals. They are found from the equator north and south, to the Arctic Circle and the Straits of Magellan, in the densest tropical forests and flitting round the barest northern buildings. Some pillaged the rich fruit gardens of India, whilst other smaller bats work hard for a precarious diet of nuts round a Siberian village. Two sharply divided groups of bats exist. One, the fruit bats, Macrochiroptera, with flat molar teeth adapted for a vegetable diet. These are found in the warmer parts of the Old World, but not in America. 2. The insectivorous bats, Microchiroptera, whose molar teeth are equipped with sharp cusps for biting their animal food. These have an almost worldwide distribution, and one species at least ranges within the Arctic Circle. The same fleas are not as a rule found on the large fruit bats as on the small ordinary bats, but some bat fleas have an extensive range. The same species has been taken from different bats of various kinds in Sierra Leone, in Madagascar, and in Java. All bat fleas are blind. This absence of eyes in fleas which are parasites of strictly nocturnal animals lends color to the suggestion that fleas which are blind have lost their eyes because they had no need of them. Disuse is speedily followed by degeneration. End of chapter 8.
Section 9 of The Flea. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Read by Larry Wilson. The Flea by Harold Russell. Appendix A. Systematic View of the Order of Siphonaphthera. Order Siphonaphthera. Latreille, 1825. Insects with body laterally compressed. Head rounded and fixed by the whole posterior part of the thorax. Mouth parts for piercing and sucking, consisting of paired mandibles with serrate margins and unpaired labrum. These are sheathed by the labium and labial palpi. Maxillae usually triangular with four jointed palpi. Eyes simple, placed in front of the antennae, occasionally rudimentary or absent. Antennae of three main segments which lie when at rest in a groove. Three thoracic segments, always free, each consisting of a notum and a sternum. The sterna of the second and third segments are further divided into the sternum, an episternum, and an epimeron, the two latter constituting the pleura. Wings and rudiments of wings entirely absent. Abdomen of 10 segments, of which the sternite of the first segment is suppressed. Abdomen enormously swollen in pregnant females of certain species. Combs frequently present on head, thorax, and abdomen. Legs developed for leaping. Coxa powerful. Femur thickened. Tarsi of 5 segments, ending in 2 claws on the distal segment. Metamorphosis complete. Larva of 13 segments. Pupa enveloped in silken cocoon. Imago, a temporary parasite, usually on warm-blooded vertebrates. 1. Family Sarcosylidae, Taschenberg, 1880. Rostrum, labium plus labial palpi, rather long, but very weak and pale, consisting of 2 or 3 segments, inclusive of the unpaired basal segment. Genal edge of head always produced downwards into a triangular process situated behind the insertion of the maxillae at the ventral oral angle. Thoracical tergites together, shorter than first abdominal tergite. To this group belong the chigos and their allies, and most truly parasitic fleas. About 14 species have been described, which can be grouped into 3 genera, viz., Dermatophilus, Hectocilla, Echidnophaga. 2. Family Pelicidae, Taschenberg, 1880. Rostrum, labium plus labial palpi, more or less strongly chitinized, consisting, except in a few cases, of 5 or more segments inclusive of the unpaired basal one. Thoracical tergites together, longer than first abdominal tergite. Here belong the majority of Siphonaphthera. 3. Family Ceratocyllidae, Baker, 1905. Head on each side with 2 flaps situated at the front oral corner. Here belong the bat fleas only. There are several genera, and about 25 species have been described. In most of the bat fleas the maxillae are shaped like a dumbbell, but in the genus Thomacilla they are triangular as in the Pulicidae. Oudemans, 1909, has put forward an alternative classification of the order Siphonaptera based on the morphology of the head. 1. Integricipita. 2. Fracticipita. End of section 9. Section 10 of The Flea. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Read by Larry Wilson. The Flea by Harold Russell. Appendix C on collecting and preserving fleas. There are 2 methods by which fleas may be preserved for study when they have been collected. The first is by keeping the specimens in small tubes of alcohol. The second is by mounting each in Canada balsam on a slide for the microscope. The advantage of the former method is that the material can be used for dissection. The student could do nothing without a microscope, though some of the commoner species can be identified with tolerable certainty by a practiced eye, which is assisted by a pocket lens. The tubes are best stored away in a cabinet fitted with wooden shelves and holes to take the tubes like a test tube holder. Fleas dried and preserved loose in a box or gummed on card are useless for purposes of minute examination and are soon destroyed. Fleas may be collected from the great majority of mammals and birds in almost all parts of the globe. They can be found in the hair and under the feathers, and also in the places where the animals habitually sleep. The best places from which a plentiful haul may often be obtained are the holes and nests in which the young have been reared. It is essential to remember, when an animal has been killed, that all the fleas leave as soon as the body of the host gets cold. No time, therefore, should be lost in searching for specimens. If the animal is small enough, it may be put into a cardboard box or a white linen bag, and a few drops of chloroform or benzene can be poured on it. In a short time the fleas will be found dead in the bag or at the bottom of the box. Some may also be found in the hairs and feathers when they are turned back. In the case of a large mammal, the hair must be turned backward shortly after death, when the live fleas may be seen running about and caught. For this purpose a small camel's hair brush is very useful. If a flea is touched with a brush of this kind, which has been dipped in chloroform, benzene, or alcohol, the insect sticks to the brush, but can be easily floated off into the tube of preservative. The best preserving liquid is 50% alcohol. Methylated spirits can be used. Acetic acid can also be used, but it is objectionable because in a short time it destroys the corks of the tubes. Each tube should only contain the fleas collected from one host, but as many specimens as possible should be secured, because there may be several species of flea on the same host. The tube must be securely corked and labeled, with the date, the locality, and the name of the host. In foreign countries it may not be always easy to do this. In such cases the skin of the host must be preserved with the tube for subsequent identification. A number corresponding to the label on the tube should be attached to the skin of the host. Fleas collected without records of the host from which they were obtained are of little or no scientific value. For this reason a tube should contain the parasites of one host only. A convenient way of preserving records temporarily is to write in pencil on a small piece of paper which can be rolled up and put in the alcohol in the tube. Small mammals generally, including bats, are good hosts. Rodents and insectivora afford usually the most fruitful captures. In trapping mice and voles, only those traps should be used in which the animals are caught alive, or the fleas will have left their hosts before they can be secured and examined. Field mice caught in the ordinary small penny mouse trap are often found dead in the morning. The best traps are made on the principle of the ordinary mouse trap, but larger. A piece of bacon rind on the hook is a good bait for almost all small mammals. Where a number of traps are put down and left out, they should, of course, be visited daily. When a live mouse or other small mammal of similar size has been captured, it may be transferred from the trap into a small white linen or holland bag. The animal can then be killed by tapping its head or breaking its neck from the outside of the bag. After this has been done, fleas may be searched for in the fashion described above. Most of the small mammals which act as hosts for fleas are nocturnal. The localities where they may be trapped are numberless, but only a small proportion of the captures may yield anything for the flea collector. I have heard of a collector of small mammals who traveled through remote parts of Spain and never lost an occasion for putting down his traps when he had to change trains at a country railway junction. In England it would, however, seldom be worth doing this, as on many lines there is an attempt to make the arrival of one train and the departure of another correspond. The following plan for securing bird fleas will be found successful. The nests of birds should be taken as soon as the young are fledged and flown. If the nest is small, it can be put into a glass-topped box lined with white paper. If too large, the whole nest, or the most firmly matted and dirty part, may be put into a glass globe, such as goldfish are kept in, and a piece of paper tied over the mouth. From time to time the nest should be slightly dampened with water. In every case a label should be put into the receptacle to preserve the name of the bird which built the nest. The bottom of the nest may sometimes be seen to be full of the larval fleas, but in any case fleas will probably emerge from pupae. The fleas will continue appearing for as much as 6 weeks or 8 weeks after the young birds have left the nest. They must be watched for and taken off the sides and top of the box with a camel's hair's brush, dipped in chloroform or benzene. I have seen dozens of fleas come from the nest of a tit, Paris major, in the course of a few weeks. They were all of the common species C. galeni. When the lid of the box is removed, it is difficult to prevent a few escaping in the room, but I have never known them to cause inconvenience to anyone. Collectors in warm countries should give their attention to the chigos and their allies, which are of great interest and have been little studied. They are found on mammals and birds in tropical and semi-tropical countries. The males are very difficult to find, but the females are large and very parasitic. They have the appearance of a small wart firmly fixed to the skin. Small mammals may be transferred, with their chigos attached, to a bottle of alcohol. Many examples of these insects are often found together on the more naked portions of their hosts. End of Section 10. End of the Flea by Harold Russell.